mentored postdoctoral fellows, 12 PhD and 8 MSc students, 11 ARS scientists, 27 scientists of ACRIC and 27 researchers from seed and TC industry, in addition to several scientific personnel research fellows. Sir has visited various countries and interacted with scientific and research organizations in Australia, China, Japan, Thailand, Taiwan and Sri Lanka. Sir is recipient of several national and international recognitions such as Most Influential Plant Pathologist in Asia Pacific, awarded by Asia and Pacific Seed Association, Bangkok, Thailand. ICR Award for Outstanding Interdisciplinary Team Research in Agricultural and Allied Sciences. ICR Award for Outstanding B.V. Mundukur Memorial Lecture Award by Indian Phytopathological Society. Dr. Harbhajan Singh Memorial Award, IIHR Foundation Day Special Award for Excellence and various awards. Sir is also a member of several national and international committees such as member of International Committee on Taxonomy of Viruses, QRT member of ICR National Institute of Biotic Stress Management, Asian Development Bank for Clean Plant Program, Research Advisory Council. He is also a member of ICR Central Potato Research Institute, REC member of ICR Central Citrus Research Institute, and member of Research Advisory and Management of Rubber Research Institute of India. Sir is currently a member of Department of Biotechnology, Government of India, Accreditation Panel of National Certification System for Tissue Culture Raised Plants, and a member of Research Advisory Committee of Merck Bioscience India Limited, Bangalore, Karnataka. Thank you.
Thank you. Good morning to all. I would like to make a journey through consequences of seed transmitted plant viruses. You all know diseases spreads in different ways, but the principal way which is important both for survival and the ecological persistence, seed is one of the means for all pathogens. But I will deal only the remote is there? You can move. Okay. Remote? Okay. If you see the importance of seed-borne virus, put key on. Plant viruses, you know, are significant crop-lossing pathogens, and their principal mode of transmission is insect vectors, and several other vectors are there, which is through horizontal transmission. But the vertical transmission is through seed, which leads to the emergence of some of the viruses. If you see the long distance transport, the principal vehicle is seed. Seed is moved internationally in different countries. So the pathogen which is existing in one location will move to the unknown destination where it is not present also. And the other problem is virus presence in the plant passing through seed is also contaminates a newer area when planting is taken in different locations. Why the virus is selecting seed as a means of transmission? It is the perfect linkage between the host and virus evolution, where they have a link which is genetically controlled both for virus and for plant. And certain plants, the presence of virus is necessary for its certain cellular processes. These viruses, what we call today cryptic viruses or endo-RNA viruses. They don't produce any symptoms, but they exist in the plant Like any symbiotic relationship we see in bacteria, viruses also have. You might have never heard about symbiosis, but there is a cellular mechanism which helps in the plants. These viruses exist in the plants. So these are no other means of transmission. It is only through seed, plant to plant propagation, or seed to seed only they will be moving. There are seeds also. One of the mechanism, RNAi, you know RNAi, which inhibits the replication or movement of a virus. Host defensive mechanism plays a significant role in transmission process. This is the current research knowledge available. Overall, the climate change environment also makes a significant change both for host susceptibility and the vector presence and prevalence and multiplication. Through this mechanism, virus evolves, and then there is an emergence of viruses. Next. Next. And if you see what are the principal means of viral characteristics that it will transmit, one of the main processes, if you see the number of viruses first noticed in 1950, there are only 2 or 3 viruses. Today, we have more than 378 viruses across more than 12 genera viruses and 7 virus families. Next. It doesn't mean that all plant viruses are seed transmitted. Only 20% of the known plant viruses are seed transmitted. And the specific interaction between host and virus plays a significant role whether the virus should be transmitted through seed or not. The rate of seed transmission varies. It is not constant. Within the host, genotype to genotype it will vary, and the percentage from 0 to 100 it can transmit. Don't have an assumption that if a virus is seed transmitted means it is transmitted all genotypes of a variety. No, it doesn't. And if it is say 100%, it is not transmitted 100% in all genotypes or on different crop species as well. This is because of the specificity of the host and the virus, which makes the certain factors, what I mentioned, RNA, which determines the how much extent the virus is able to survive when it is passing from cellular process, from endosperm to from seed coat to the embryo. In all these processes, when it travels a journey, it determines where it can able to move in all the passes, it is able to successfully transmit. But in the process, hosts also have a defenses which restrict the virus. Some of them restricted only on the seed coat, some are in the endosperm, some in the embryo tissue. Next. And if you see the passes, what I mentioned, ovary wall is the one, certain viruses penetrate in the ovary wall, and certain viruses transmitted through pollen. Then certain viruses exist in the meristematic tissues, will move ovule or ovary, then it reaches to the gamete formation, and it is the embryo developing, it retains in the embryo, and the embryo when it is germinates, when it is growing, it will transmit to the plant. So any transmission mechanism, when we say it is a seed transmitted, it has to pass from plant, seed to plant, then only it is seed transmitted. Otherwise, it is seed contaminated or seed mixture. Next. Then the next, seed borne, 2 types of seed associations. One is externally seed borne, where the presence of a virus, it is on the surface of the seed coat. The another one is the internally seed borne, means when it is passes through seed coat to the endosperm and then embryo, if it retains in the embryo, then it is internally seed borne. Next, if we see the location of seed contamination, most of the time it is not a biological significance association, it is a mere contamination on the surface. So the virus is attached on the surface of the seed coat. It can be easily removable where we follow the seed treatment methods, disinfection methods, which helps such viruses can be easily removable and which are seed contaminant ones. Next page. And if you see the modes of transmission, as I mentioned, when it is passing from horizontal transmission is done through plant propagation and insect vectors, nematodes, fungi, all these are the vehicles it picks from the infected plant to subsequent spread. But whereas vertical transmission, the virus can move in the pollen or in the embryo. When it is there, then only it can be propagated from plant to seed and seed to plant. Next one. The three mechanisms what I am mentioning in a viroid and a virus, the similar approach is there, where if the virus is present in the entire gamut of the tissues, the meristematic tissue, you see first line A, completely the color it is, entire growing bud is infected, virus is there. When from such thing growing, the gamete is developing, the embryo is developing, it will show symptoms on cotyledon, seed coat and embryo. It is true seed transmission where it is from embryo to it is transmitted and complete. It is the responsible for vertical transmission of the viruses. The second one is where the meristematic tissue, as I mentioned, it is restricted, but it is mesophyll tissue, it is present, and when it is present on mesophyll tissue, it can come on the endosperm or in the cotyledon, but it is not passing to the axis, embryo axis, so it is no vertical transmission. Whereas if it is passes through the embryo means it is vertical transmission. The third case is pollen transmission, wherein the virus enters through pollen, which is a female ovary. When it is combined, the virus present in the pollen will enter into the embryo, then also it will lead to the vertical transmission. The normal mode of survival of plant viruses, as you all know, it is the initial, if a plant is healthy plant, if it is infection comes from an insect or through seed, it is a survival mechanism for the viruses. It can survive in the seed or planting material or in the insects. Then it will go and attack on a healthy plant and produce symptoms. From such symptomatic plants, again, insect vectors will pick, transmit to healthy plant. It is also a pollen which are certain hosts. It is also pollen transmitted. Viruses will carry in the pollen. The emergence, one of the best example currently known is tomato brown rugose fruit virus. It is externally contaminated. Initially first noticed in 2015 in Jordan and Mexico and Israel. 3 places simultaneously within 2 years it is recorded. Now this virus is present throughout the world. There is no continent which is not present. Majority of the countries, it is mainly infecting earlier in tomato. Now it infects pepper, chili and hot pepper and sweet pepper and brinjal, and also it is infecting other leguminous hosts. Currently it is going even legume hosts. And initial notice, you see the spread in the entire world. Because of the seed movement, the virus was existing in 3 locations, has spread to the worldwide, and in different locations it is called symptoms and disease expression is prevailed. It is emerged as a world pathogen and there is a quarantine restriction for this virus for worldwide. lethal necrosis. Very serious concern in African countries, and it is also spreading in America. In India, Asian countries, it is not yet there. As it is seed-borne, at any time it is possible to prevalence in India as well. Similarly, tobacco rotting, papaya melia virus, which is very prevalent in Brazil, but in India it is not there. It is also passes through seed. If we import such seed and not tested, we are likely to get this papaya melia virus as well. There are certain pathogens, no insect vectors are involved, or nematodes are not involved, but they are highly transmitted through seed only, and some are pollen associated. Some of them, apple chlorotic leaf spot, which is very common now, very high incidence even in India, apple, and in citrus. It is present in citrus as well. And barley stripe is again in maize, barley, and wheat. Melon necrotic is a spot in cucurbits, melons, and watermelon and muskmelon. It is very serious, especially in Southeast Asian countries. Now even it is there very much in India, and it is a quarantine restricted virus. Pelargonium donate spot is again common in pepper and tomato. They are all pollen or seed transmitted. Pepinomosaic is a potex virus, is again seed transmitted. And southern tomato virus, it is also, symptoms are not expressed in certain genotypes. It is highly seed transmitted, only passes through seed, no insect vectors involved in this. And tomato bush resistant virus also, no insect vectors, it is in the soil water movement, or whenever nematode is feeding, its presence in the water and the soil will come in contact with the cells, damaged, and enters, and that it becomes established. But there is no mechanism of vectors transmitting this. The classical examples of seed contaminated viruses are trabemoviruses. There are cucurbit infecting ones, cucumber green mottle, Cuslias green mottle, zucchini green mottle mosaic virus. These three are in cucurbit, all cucurbit crops, this is very prevalent. And pepper mild mottle virus, which is only present in peppers so far. And tomato brown rugose, as I was mentioned, its seed transmission, true seed transmission doesn't happen through embryo. It doesn't infect embryo, but it is there in cotyledon, and that cotyledons, when during germination, the soil particle or damage happens in the axis or cotyledons, this virus present on the seed coat will enter and establish. That establishment is very less number, 1.8 to 2% only. But it is highly contagious, so contamination from seed to seed lot is the common means it spreads. Same thing, tomato mottle mosaic virus and tobacco mosaic virus. All belongs to trabemovirus. These viruses we have done extensive testing, several of the imported germplasm and seed. We found tomato mottle mosaic presence in whole seed, seed coat, but cotyledon, but not in the embryo. It is only surface contaminated. Sometimes in some genotypes you will find in cotyledon, but not in the embryo. And we also developed a multiplex PCR for these trabemoviruses in solanaceous crops because they are quarantine issues. Whenever seed is to be exported, we have developed multiplex PCR for all the 4 viruses. The presence can be detected using 4 set of primates. And seed transmitted viroids is another important pathogens which normally is through seed to seed only they transmitted. They are pollen and seed. There, that is why you will find 100% infection certain crops, they are viroids. Initially they don't express symptoms, only under certain environmental conditions they express severe symptoms. Otherwise, most of the same symptomlessly they are carrying. And they are all now currently serious issues for seed export and seed exchange. Any solanaceous host or cucurbit host, without testing with viroids, no country will accept. these viruses, earlier we have seen few viruses and now we are seeing so many viruses. The major advantage is because the detection technologies, diagnostics developed currently, more sensitive diagnostics are there. Earlier we were using only physical methods, electron microscopy, and then bioassays, inoculations, which is time taking and it is not possible to detect. But currently now we have moved from antibodies to the molecular assays, which are more sensitive and faster. Large number of samples we can test. Because of these tests, we are now able to detect several viruses present in the seed. Even the current technology, high throughput sequencing or next generation sequencing, has made possibly in several hosts, new seed associated viruses have been detected. As I mentioned, endoRNA viruses and cryptic viruses, more viruses are detected now because of the NGS technology. But all these techniques, for a regulatory purpose, diagnostics are different. For laboratory testing is different. If you see confirmatory field of diagnosis, you are using ELISA or PCR. For quarantine purpose, bioassay compulsory for tobamoviruses. ELISA, PCR, quantitative PCR is compulsory for tobamoviruses, especially tomato brown rugose and tomato mottle mother virus. And this is how the tests, when the seed arrives, seed is tested using a PCR or ELISA, and then with sensitive techniques also, if the germplasm is to move, the next generation sequence is used to confirm no virus is present in the seed bar. Some of the methods are now internationally made regulations wherein how much quantity you have to take for testing a seed. If I am exporting 1 ton, can I test 1 kg, 1 gram, 1 seed? How it is to be done? There is a standard developed for any test to be done. ISTA and ISI has, and US National Seed Certification Agency has developed 3,000 seeds per lot. Each batch of 250 seeds has to be taken for some subsample. Minimum 12 subsamples are required to be tested in duplicate, either in ELISA or PCR. If you do that much number only, you can say less than 0.1% infection is there, you can determine. Otherwise, if you test 100 seeds, then the acceptance is not there in quarantine. Similarly, for viroids also, we have in QOPFR, because of international importance, emergence, they have put 2 assays compulsory. Initially, we have to test bioassay. If symptoms are there, then it has to be rejected. If symptoms are not appearing, the seed has to be grown and seedlings has to be tested with qPCR, quantitative real-time PCR. If that test says no, then seed is accepted. Otherwise, seed is not accepted. In case of cucurbits, again, seed standards are there. The minimum number of seeds here is not 3,000. It is 2,000 seeds, 20 subsamples, 100 seeds is to be tested in one sample. Viroids also similarly. Viroids also like tobamoviruses. 3,000 seeds are required to be sampled. Each subsamples of 6 to 12 subsamples consist of minimum 500 seeds per subsample. This is how for viroid sampling has to be done. And we have done STS for chili crop seeds for testing, and the seed collected from showing symptoms of leaf curl type of symptom. We thought it is only leaf curl virus may be there. We wanted to know whether other viruses. When we tested, we found more than 6 viruses present in that. 2 of them are endoRNA viruses. One is cryptic viruses. And in addition, common viruses like chili winter mottle virus, tomato mosaic virus, and chili vinyl mottle and cucumber mosaic, all three viruses also present in the seed, but were not in the embryo. They were there in the cotyledon only, in the seed coat, but not in the embryo. For any diagnostic applied, the purpose, for lab purpose, I told you have to use molecular test. If it is quarantine, PCR, bioassay and PCR also two types, qPCR and then normal PCR for certain viruses. Because of this is the country to country requirement, it is depends. If the seeds are transmitting, how to manage those things? When we say so much, if seed is coming through sweet, how can we stop this? Yes, we can stop certain extent, it is not 100%. One of the means is the disinfection. Disinfection means the viruses which are present on the seed surface, we can treat with different modes of methods, which are chemicals, mechanical, physical, chemical, biological, and then other methods. In all these cases, the surface-borne virus particles can be easily eliminated by all of these methods. But whereas internally seed-borne cases is possible only two methods. One is the plasma, cold plasma treatment. Another one is mentioned, that is also reduced. 1% or 3% sodium hypochlorite initially used, but what was not subsequently found effective in a large scale seed is used. Then there are products, Viricon S, Minon fluoride, Jet 5, all these chemicals are found very effective in reducing the tobacco. If you see Florax, Viricon S, NFT, even skimmed fat, non-fat milk also is used for treating certain pepinomosaic virus, virus and TME can easily be removed by this method. Whereas TOBFR, these are not easily removable. So Florax and Viricon are effective for TOBFR and also spindle tuber viri. The type of plasma I was mentioning, plasma is a method wherein you expose with a certain exchange gas, there it emits plasma cells into divide gases into oxygen, hydrogen peroxide, nitrogen. There so many radicals will evolve. These radicals interact with the virus replicative process and inhibits the virus. Some of the tried ones are TMB, classical example, they have done and found completely virus could be eliminated. Biological treatment, you all aware that today microbes, fungus, bacteria, trichoderma and pseudomonas, bacillus, we are extensively using for fungal and bacterial disease. But it can be equally applicable for even viruses. We may think how it is acts, but there is a mechanism, it is the signal transduction mechanism wherein it stimulates the host mechanism to suppress the virus multiplication, and some of them I am giving which are already proven in the field conditions, crops, CSW, TMV, chili vinyl mottle virus, pepper mottle virus, cucumber squash mosaic, Duchney yellow mosaic, bean counter, all these viruses has been tried with different bacillus formulations used in the seed priming and subsequently reduction in the virus population, or some cases totally inhibiting the virus multiplication. Finally, it is the methods which you have applied is also starting from the crop growing conditions, selection from the seed, multiplication, seed production conditions we need to harvest, avoid infected plants, then selecting healthy, vigorous growing plants for seed, and then that harvesting process, grinding, then packing, after finally pack the seeds to be randomly tested and confirmed, no virus presence, certified as pathogen-free seed material. So for a reliable diagnostics, advanced diagnostic application is very critical for seeds, and such methods to be applied based on the requirement, and healthy crops produced will give higher productivity, crop enhancements which help in the farmer and increase in our agriculture production. That's all. Thank you. If there is any clarification questions, yes.
Thank you, Dr. Reddy, for the excellent presentation. I just want to see one slide where you mentioned the germplasm has helped in distribution of viruses.
Yes.
I just want to see once.
Oh, slide you want to see? Slide. Down. Down. Faster. Down. Up. There. That is.
And I just want to know, like, where from this information has come.
This is the recently compiled a paper, the seed movement and virus exchange. It's a slide from on paper.
Okay.
They have compiled, see, they have made a data, and you see, do not necessarily depict actual location.
That is what I.
No. But it represents exact location, means that map exact country location may not be appropriate, but in that country it is there.
But it doesn't mean that the germplasm has contributed for these viruses, no?
Yes.
I just
See,
want-
that data they collected from the base information. Suppose in our country, TOBFRA we have reported in 2024. That we have reported from seed received from Netherlands. So if that seed is grown in that area, it is come, I am publishing that information is put in it as an input value. That is how it is the published information through imported germplasm where the new virus is reported, such recording.
I would like to have that back.
Yeah, I'll send you that review paper.
Okay. Thank you.
Dr. Reddy.
Yeah, sure.
If the temperature, one of the slide you showed, if the temperature is 70 degrees Celsius,
Yes.
for, you can look at here,
Yeah.
you can ask.
Enough?
70 degrees Celsius,
Yes.
you may eliminate virus, you will eliminate seed also, I think. Viability.
Exactly, 100% you are correct. The seed is tried in from 35 degrees to 70 degrees. Some viruses 35 to 47 degrees is enough. Like tobacco viruses, TOBFR. Even at 70 degrees it was not eliminated. Seed viability is also lost. But in cereal crops, barley stripe mosaic, at 70 degrees heat is not continuous. It is heating, cooling, heating, intermittent cycle. When they have done 70 degrees cycle with different intervals, that has been reduced because without affecting germination. But it is in cereals, germination was not affected, whereas in cucurbits and solanaceous host, seed germination is drastically affected. It is not in all crops. It is crop specific and virus specific.
If you take sugarcane sets,
Yeah, it is helped in sugarcane up to 47, not beyond 47.
vegetatively covered.
Yeah. Sorry.
Sir, I have a slide. This is actually you are relating the epidemics with the introductions. Sir,
That is one of the factors.
one of the factors. But
Yeah.
I am seeing a trend there. Look at the map. everything is in the global south.
Yes.
You see, of course, the diversity is actually existing in the global south only. And the diversity, I mean, lot of material movement happened mostly, within the global south. I think if the epidemic is related to the introduction of a particular material into that global south countries, I think most of them are the Latin American and including Asian. So you can actually, if it was not there earlier, why not you conclusively say that this happened? You are only trying to say that it is attributed, but you are
No,
not.
not attributed.
Yeah.
It is actually recorded evidences.
Thanks very much.
These are all recorded evidences.
Sir, this tomato spotted wilt virus was reported in tomato only or any other crops?
No, it is not even tomato. It is in pepper.
Pepper.
China 2022 they reported.
From
in
India,
pepper.
anybody reported? Sir?
No, India we have tested several times. Not only TSWV. India most common virus is groundnut button crust.
But recently from TNA,
Oh no, wait. I will come to your point. See, we have tested the bigomovirus TSWV and all other bigomovirus present in India. Several seed samples we have tested. On the seed surface, seed coat you will detect, but seed transmission doesn't happen in Indian viruses. Then tomato spotted wilt virus, I have not yet published, we have detected in capsicum, my colleague detected in tomato, and me and that Selvaraj detected in a medicinal plant. TSWV, which was not noticed till 2018 in India. 2018 we have noticed from chrysanthemum, imported cuttings brought to Tamil Nadu, multiplied, then it is prevailed. From that time onwards, we are now noticing in tomato some ornamental crops. But seed transmission we have not detected so far in Indian countries.
Okay. So not extensively tested in.
No. First virus itself is not extensively.
It is testing by NGS approach or by PCR?
We tested by PCR. We tested one sample by NGS, but we got GBNV, but not the TSWV.
Okay.
TSWV we didn't get in the seed.
Okay. Thank you.
Again, extending to that first question, it is in Madanapalli area. I am from Coromandel, from Chakrada. So we are doing a lot of extension service. We observed kind of farmers say it is jaundice. So fruit, it is tomato spotted wilt virus.
No.
It's not.
No. It is not tomato spotted wilt virus. This problem we started in 5 years back in Maharashtra. We monitored there continuously. Now we have given the package how to develop. It is cucumber mosaic virus which comes. Cucumber mosaic virus is known to produce symptoms on the leaf, but you don't see symptoms on the plant, but you get fruits yellowing.
Yeah, fruits yellowing.
Uneven ripening.
Uneven ripening is
Yes, this
all
is the,
commercial hybrids are
Yes.
susceptible for this.
to mention that only, virus is emerging, modified. It is more expressing in reproductive organs, but not in the photosynthetic area. It is evolving. So this isolate which are strain, which is infecting in Maharashtra, we have studied extensively. It doesn't produce symptoms in the plant. Initially for 4 years when we are continuously monitoring, no symptoms in the plant. You see, it is a healthy plant. Fruits
Yeah.
you will get yellowish.
But mosaic has to show some.
Now it is showing mosaic also. Slight mosaic symptoms are appearing now. You will see these symptoms only in plants which are sown in January, February, March.
Exactly.
Months only. In a
It
rainy
shows
period,
expression, yes,
rainy
exactly.
period sown
Correlating.
crop, you don't get this symptom or you don't get the virus.
Exactly, sir. It's correlating. You are
Yes, that is, we have studied this in Maharashtra. Now every year before tomato planting, I go to the Maharashtra farmers and give 1 day seminar for them how to manage.
And how here also you can do the same answer in Madanapalli area.
Yes, I gave one lecture in Madanapalli. Subsequently, last year I gave. My principal method to communicate them is first nursery people. This disease mainly comes from nursery people.
Any ester molecules are to initiate any host resistance in the molecule?
Yeah, it is there. You can apply the seaweed extract. You can suppress very much.
Thank you.
Yeah.
I think Dr. Reddy made an elaborate presentation. We shall further continue during our lunch hours after post-lunch session. We should move on to the next speaker.
I thank organizers, especially Honorable Vice Chancellor Dr. Joshna and Pradeep, asking me for this lecture to come. I asked them what I should give. They were asking for re-emergence virus diversity. I told virus diversity doesn't mean anything. Let us speak seed so that people should understand how it is forming. So I choose this topic to convey that we need to increase awareness. Common phenomena, anybody, even in the policy making. Seed transmitted means any seed is seed transmitted, any seed virus is transmitted. It doesn't happen. So we need to test. It is the genotype dependent. It is not same amount of percentage it won't transmit. That is what I want to convey. We should not be panicked it is seed transmitted, but we have a method to assess and do the management. Thank you, one and all.
Thank you, thank you. I think we should go to the next speaker, Dr. Celia Chalam. I think she is being introduced by the organizer side.
Good afternoon, everyone. Now I would like to introduce Dr. V. Celia Chalam ma'am, Head, Division of Plant Quarantine, ICAR, NBPGR, New Delhi. She did her PhD in plant pathology from Acharya N.G. Ranga Agricultural University, and she has more than 30 years of research experience in plant quarantine, diagnostics of plant viruses and phytoplasma, policy issues related to biosecurity and biosafety. And ma'am also screened more than 4,500 accessions of sesame germplasm against phytoplasma under field and artificial conditions and identified source of resistance. And ma'am spent 4 deputations at Washington State University, USA, and Oklahoma State University, USA, Mudroch University, Australia, and IITA, Nigeria. And she has received D.P. Mishra and R.N. Pandey Award, Best Women Scientist Award in 2023 by Indian Phytopathological Society for outstanding contributions in plant pathology. And ma'am also received Dr. R.D. Vijay Prasad Rao Memorial Award 2012 for outstanding contributions in diagnostics and plant biosecurity. And ma'am is the fellow of Indian Society of Plant Genetic Resources and fellow of Indian Phytopathological Society. And she is an expert member of international committees and national committees as well. And she organized 30 HRD programs on biosecurity and biosafety for plant quarantine, custom officials and scientists from private sector and public sector. And she studied biosecurity systems in other parts of the world during participation in various international conferences in Australia, USA, Europe, Africa and Asia. And she published 80 research papers, authored 6 books and 12 manuals and more than 25 book chapters, 20 technical bulletins, and presented more than 150 papers in international and national conferences, including several lead papers, and delivered more than 100 lectures in training programs. Now I would like to invite ma'am to deliver a keynote address.
Thank you. Good afternoon, everyone. I would like to thank the organizers, especially the Honorable Vice Chancellor, Dr. Sarada Jayalakshmi, Dr. Jyotsna, and other committee members for giving me an opportunity to speak here. The topic is National Plant Quarantine System for Crop Biosecurity against exotic pathogens. The previous speaker, Dr. Krishna Reddy, has set the tone for quarantine, because in quarantine seed transmitted is important, and even if it is seed borne, if it doesn't go to the next generation, it is not of importance for quarantine. Thank you, Dr. Krishna Reddy. The next, yeah. Yeah, why do we talk about the biosecurity? Like there is a transboundary movement of the planting material either for commercial purpose, either for planting or even for consumption, and the germplasm which cross the borders for crop improvement programs. And the commercial material, because the volume involved is heavy, so the risk of introduction of pests is there. And equally the germplasm, if it is coming from USA, the pests which are occurring in the USA can come along with it. And in my talk, if I am using the word pest, as per the International Plant Protection Convention, the pest means anything, fungi, bacteria, viruses, insects, mites, nematodes, even the weeds. And internationally, there are several examples of global spread of pathogens during the trade. Like the today morning, the Vice Chancellor has mentioned about the Irish famine, where millions of people died and millions of people migrated to USA from Ireland because of the Phytophthora infestans. And if my former head of the division is here, Dr. Ravi Ketrapal, some of you know, he says that Abraham Lincoln has become the President of America because his family migrated from Ireland to USA because of Phytophthora infestans. And there are many examples, like the wine industry in France has wiped out because of the introduction of downy mildew, powdery mildew pathogens into France. Can you help? Just look at me, I will say like this, then you go to the next slide. Next. And these are some of the recently introduced exotic pests. Like this is basically the students. It is very useful. Like since 2015 onwards, there are many insects, and the famous one, follow me worm, everybody knows. And now the private industry and the public sector started working on varieties resistant to follow me worm. And also the famous black thrips in Andhra Pradesh, it has caused devastating damage in chilies. And the walnut leaf miner in 2024 in Kashmir. If you go to Srinagar, you will see this. Then you might ask, then if there are introduction, what are you doing? See, if there is no quarantine, there would have been many more introductions. Next. You have 1200 pests which are regulated. If you look at it, like there are 500 insects, 264 viruses, there are many fungi, they are yet not reported from India. The previous slide, there are unintentional introductions, especially the insects. How do they come? They don't come with the seed or any seed, but it is possible with the planting material, but they travel miles and miles. And my structure of the presentation is, in the first part I will talk about what do we do in quarantine, and also I will try to touch upon some molecular diagnostics and genome assisted tracking of global movement of pathogens and artificial intelligence. So to ensure biosecurity, what do we need? We need the regulations, both international and national, and we need the diagnostics which are reliable, sensitive, specific, and also we need the capacity building. Suppose if you have the regulation, you don't want the bean pod mottle virus not to come into the country. If you don't have the diagnostics, how do you stop it? Or you don't want the bean pod mottle virus to come into the country, but you don't have the regulation. Your national regulation doesn't mention that, then also you cannot stop it. And also capacity building. When in initial stages of my career, I was in IHR Bangalore, and we used to get the germplasm tomato chili from World Vegetable Center, Taiwan. I have no idea like what happens in NVPGR. I used to think that the material is coming directly from Taiwan to me. So and also the researchers also should be aware that you should not be bringing the material. If you find anything important, like some of the things like hing, people bring from Afghanistan, Iran, and then come and ask us, we got the material. You have already brought in, what do I do with the quarantine? So that is also equally important. And customs, if you coming from overseas, whom do you see? You see the customs, they have the search and seize powers, not the quarantine. So the capacity development of the awareness among customs officials are also equally important. Next. And these are the international standards for phytosanitary measures. There are 47, but the ISBM 30 has been repealed, so now we have 46. And how to develop the disease-free areas, or how to report a pest, or how to do the sampling, how to have the post-entry quarantine station. I have not mentioned all because of paucity of time. And this is the national plant quarantine system in India. All over the world, India is the only country where we have the biosecurity quarantine for commercial material, one agency deals with it, and germplasm quarantine, another agency deals with it. And bulk material, which is for commercial purpose, is dealt by Directorate of Plant Protection, Quarantine and Storage. We have 73 plant quarantine stations across the country wherever there are seaports, airports and land frontiers like Amritsar, Wagah border. And the other side is germplasm, where I come from, National Bureau of Plant Genetic Resources. We have the Division of Plant Quarantine at headquarters Delhi, and also we have it in Hyderabad, which look after the quarantine of germplasm meant for Icresat, and also southern part of the country. And there are different schedules under the PQ order. I will not go into the details. If you just type the plant quarantine order 2003, you have the 340 pages of the plant quarantine order you can download, which is free. And this is just one example where in Schedule 4 we prohibit the material import. For example, wheat is prohibited from Latin America and Bangladesh because we have recently found wheat blast in Bangladesh, which is adjacent to West Bengal and borders are porous. So the Indian government has prohibited the import of wheat from Bangladesh recently. Yeah, this plant quarantine order 2003, you may think that it is more than 23-year-old. No, this order is 2003, but there are amendments which comes from time to time. If the crop is not mentioned, if the country is not mentioned, the amendments come in. There are about 146 amendments in these 23 years. And for example, you can see here in 2023, the apple fruits from Germany is not mentioned, though the pest risk analysis was done and the pests A2W, almost more than 20 pests are mentioned here. So that this draft notification is put on WTO website and then it comes into existence. And also the Indian government has the standard operating procedures for post-entry quarantine. And post-entry quarantine now, because of the COVID, we don't have to explain it much. And these are the post-entry quarantine, just a glimpse of it. Otherwise, 222 crops post-entry quarantine is mentioned in the plant quarantine order 2003, where it is a high-risk propagative material. And these are anywhere between 1 to 2 years to 5 years for coconut, and for wheat it is 1 season, and for potato 2 growth seasons. And for post-entry quarantine facility, both closed facility and the soil-based open facility are acceptable. And then unlike the greenhouses used for basic research, all the vents should be covered with 40 mesh net, and there should be double door entry. Even the cold cellulose pads and the fans area should be covered by 40 mesh net so that no insects will come inside. And open facility doesn't mean that it is completely open. It should be 500 meters, half a kilometer to 1 kilometer distance. There should not be any same crop or similar crop. Suppose if you are growing wheat, there should not be barley, triticale also, maize also, because the pests are same. And this is the one where in open facility you should have 10 feet height, you have the dense polythene, and then proper gate, locked. And these are the list of inspection authorities. Even in Andhra Pradesh you can see the Angrao, Guntur is mentioned. And if the head division of plant pathology cannot undertake the inspection, he can nominate the pathologist and entomologist. And recently in 2023 December, head division of plant quarantine and officer in charge regional station Hyderabad are gazette notified as the inspection authority, so that we even certify the facilities. Yeah, and for the commercial material export certification, there are almost 200 phytosanitary certificate issuing authorities, Gazette notified, and follow the ISBN 7, 12, and 23. And now for the house, there is no export-import committee. Exempt committee has been disbanded since last year. And then there is an online system where both customs and quarantine it is a single window system. Earlier, the applicants has to go to exempt committee. Now it is disbanded. And the Indian government has also developed national standards for phytosanitary measures. As per the international standards for phytosanitary measures, there are 24 national standards phytosanitary measures, and the number 23 guidelines for phytosanitary inspection agency and phytosanitary inspectors. This is a good movement for public-private partnership. And there are standard operating procedures developed by the government of India. Even like we export a lot of rice to USA, China, there are standard operating procedures. We export a lot of vegetables and fruits to EU, there are standard operating procedures. And even for post-entry quarantine inspection, I already mentioned. And for plant quarantine import clearance also, there are standard operating procedures available. This is a very good example of public-private partnership where phytosanitary treatment providers are accredited by the NPPO, that is DPPPS, and also the registered pack houses processing units are also approved by the NPPO. And the entire certification activities are carried through the PQMS website, and only the importer has to register himself, and then he can even track where the samples are. And then the customs people, and there is a coordination between customs and quarantine through this online. And till this is about quarantine for commercial purpose. And then the germplasm quarantine director and DPGR is empowered to give the import permit, and then for export material, Division of Plant Quarantine head gives the phytosanitary certificate, even the OIC regional station Hyderabad. And transgenic germplasm is quarantined only at Delhi, not at Hyderabad. And during the last 5 years, if you look at it, we have quarantined about 7.5 lakhs germplasm samples, and we have exported around 2 lakh samples of germplasm. And phytosanitary certificates, there are 294 phytosanitary certificates were issued during the last 5 years. And also we charge whatever we do per sample, we do the quarantine processing charges. And more than 100 pests were intercepted during the last 5 years. And if you look at it, almost more than 40 pests which are not reported from India, we have intercepted in the imported germplasm. And this is how the entire procedure done. All the scientists, technicals, there is no sampling here. Each and every sample is inspected, and then we remove the discolored deformed cells, and then it will go to different labs. And also in the transgenics, we test for the absence of terminator gene, that is embryogenesis deactivator gene. And all the legume germplasm is grown in our facilities, and the rest of the germplasm is provisionally released. And if we don't have any phytosanitary treatment, then we incinerate the material, and only the pest-free material is released to the inventor. And this is the joint inspection where all the scientists and technicals examine and remove all these discolored seeds. And since this is a biosecurity, I cannot talk only about the pathogens. It is a comprehensive one. If I say, okay, fungi, bacteria, viruses, what about insects, nematodes, weeds? So I have a few slides for diagnostics for these pests also. And we do the real-time X-ray radiography where the seeds looks absolutely healthy, but we expose them to the soft X-rays. You can see the dots here. Those are infested by blowpits and chalk seeds. And also we do the seed transparency test. And these are some of the just mentioned as an examples, but we have intercepted many more. And here for detection and identification of fungi and bacteria, we don't undermine the taxonomic abilities using the microscope. We really honor that. But besides that, we use the molecular techniques also like PCR, loop mediated isothermal amplification, LAMP, and other techniques. And these are some of the fungi which is not reported from India. And for nematodes as well, we do the taxonomic identification, morphological taxonomic identification using microscope and also the molecular techniques. And weeds, we have a weed scientist, and these are just an examples of exotic weeds intercepted in the exotic germplasm. And the legume germplasm of soybean, fava bean, French bean, pea, cowpea, all this germplasm is grown in our own greenhouses. Like there are out of 130 seed transmitted viruses, 50% belongs to these legumes. That's why these legumes we don't give it to the inventor. And then all the symptomatic and also the healthy looking samples we test for viruses using electron microscopy, ELISA and PCR. During the last 5 years, we have intercepted 15 exotic viruses in the germplasm. And all our scientists, we undertake the post-entry quarantine inspection. As I already mentioned, it is a provisional release. And if any one of you is importing the germplasm, please do not use it immediately. You need to wait for the quarantine clearance. Yeah, these viruses you can see whether it is from USA or Netherlands or Taiwan. We have intercepted even African countries like Mali and Niger. These are all viruses not reported from India using, I already mentioned, ELISA, PCR, and we have multiplex PCR protocols. Yeah, the same testing we do for transgenics. Like during 1997 to till date, we have imported more than 16,000 samples. Only thing is the clearance from Review Committee on Genetic Manipulation from DBT Government of India is required, and the material is processed in containment facility. You see here, this has high efficiency particulate air filter so that no pollen or no pathogen will go outside this facility. And transgenics doesn't mean that they come clean. No, they also, we have intercepted many fungi, insects, and even the viruses in this germplasm. Because I'm just giving one example of transgenic soybean imported from USA. We have intercepted 5 viruses not reported from India. It is not intentional. Don't think that it is some kind of agroterrorism or bioterrorism. This is, they are giving the germplasm and we are doing the strict quarantine and intercepting the viruses. And these are all the phytosanitary treatments which are adapted, and like mechanical cleaning itself is a phytosanitary treatment. When you get a fever, what do you take? You take the paracetamol to kill the virus, bacteria and all. So the same way we do the disinfection, disinfestation, and hot water treatment is given to rice. It is a prophylactic, 52 degrees centigrade for 30 minutes. Here it is tested that viability is not affected at this temperature. Acid wash, spirit wash, and X-ray radiography itself is a detection and salvaging technique. And fumigation is given whenever there are any live insects or eggs are found. And after this entire quarantine procedure, all the plants are uprooted and disposed following the standard operating procedures and guidelines. And we have also developed the guidelines for import of transgenics. If any one of you is interested, we can send the soft copy. And if depending upon the state pollution regulations, if they allow, then we incinerate the material. Incineration is simple burning. Or if the state government has certain rules, they don't allow the burning, then we dig a pit and bury the plants. It is almost similar to the burying a dead body. And this is an analysis we have done, like the impact of regulations, diagnostics. What if we have not done quarantine? If we have not stopped the entry of these pests into India, they could have come, they could have got established, they could have got spread. So, if we have not done that, for example, in soybean, if I have not intercepted bean pod mottle virus, or in maize, soybean downy mildew, so it would have caused this million rupees of loss. Like in soybean, 261 million rupees loss we could have caused, and this paper is under review, we would be publishing soon. So by closely monitoring which pests are mostly frequently reported from which part of the country. For example, when Bangladesh we have found the wheat blast, then the Indian government has immediately banned the import of wheat from Bangladesh, and also the research was conducted for identifying the resistant sources, and also so that we can predict high risk pathogens, commodities or region. The best example is wheat blast in Bangladesh, and also the collecting and disseminating the information across the Asia and Southeast Asia region. Like I always say this because I have traveled that part of the country, world also, in Southeast Asian regions. Like if you go to northeast part of the country, you can go to Myanmar, from there you can go to Thailand, you can go to Vietnam, Laos and all. So the borders are also porous. So there should be some kind of network between India and these Southeast Asian countries. So that second part, I'll take 10 minutes more, maybe less than that. The molecular detection techniques, why do we talk more about it? Basically because lack of taxonomies. The younger generation is more into molecular diagnostic than the morphological identification using the microscope. And there are many seed-borne, seed-transmitted pathogens, fungi, bacteria, viruses, and also if you have a saprophytic fungi, it masks the actual pathogenic fungi. So that is also one of the reasons we need to have. And also if you are reporting a new fungi or new virus, you need to conform with the molecular diagnostics nowadays. You just simply say that this is a fungi that doesn't stand. And of course for rapid detection of pathogens, and also you have the automation like PCR machine. You mix all the reagents and put it in the machine, and the machine does the job. So that is also one of the things, like we need molecular detection. These are all the techniques. So in our division we have developed the IDS based molecular detection of soybean Peronospora manchurica, which is not reported from India. If at all it comes along with the germplasm, we use this molecular diagnostics for detection of soybean downy mildew. And this is for identification of Xanthomonas campestris, Pathover campestris. And you may be saying that it is reported from India, but it is a regulated pathogen as per the PQ order 2003. And we have many simplex PCR, multiplex PCR protocols for viruses not reported from India. For example, bean pod mottle virus, not reported from India, grapevine fan leaf virus, tomato leaf spot virus. And this is a multiplex PCR for detection of 5 quarantine viruses. Since our lab is entitled to work on the quarantine viruses, whenever we intercept, we use them as a reference material. So we have developed the quantaplex PCR protocol for detection of 5 viruses in soybean. And this is 4 fungal pathogens of soybean in quarantine. And these are some of the techniques for real-time RT-PCR and HDA, helicase dependent amplification, loop-mediated isothermal amplification. Of course, this is part of the work I have done when I was in USA, high plains virus. And this is the reverse transcription recombinase polymerase amplification assay for detection of peanut mottle virus in quarantine. This is the work done by Dr. Parmeshwari. And like even the deep sequencing, whole genome sequencing can be used in quarantine. This is just one example of the work done elsewhere in the world for detection of seed-borne viruses. But using the deep sequencing, NGS, all this it is not economically feasible in quarantine unless and until it is a high risk pathogen and high risk commodity. So these are some of the examples where genome assisted tracking of plant pathogens movement. When I saw Dr. Krishna Reddy's world map with all the viruses, I was thinking somebody has to do actually the like this, like UG99, how it has come from Africa to Middle East, and then citrus canker and HLB disease, and even the Panama disease, tropical race 4, affecting Cavendish bananas, and there are many examples. Like this is one of the well-studied disease where genomes assisted global movement tracking was done for wheat blast pathogen. When it has come from Latin America to Bangladesh, then immediately, but that it is a global project where findings were shared on an open read website, and genomic data serve as the foundation for point of care disease management. Like this work was done by Islam and his other scientists in Bangladesh, and they have done it within 6 weeks, but a lot of budget was involved. And genome sequencing of 537 worldwide distributed Magnapodia oryza isolates were carried out to know the distribution of wheat blast, and finally it was attributed to the Zambia one caused by B71, the Bolivia one. So this is actually the genomics, how they are used to track the movement of plant pathogens. And this multi-locus sequence typing also was used for Ralstonia solanacearum, and then it was the lineage of this bacteria found in India which has exhibited similarity with US and Europe. And even Xanthomonas oryza, Tatavar oryza, the lineage of African and Asian strains was established, and the one which was introduced into India was tracked to other Asian countries, possibly through the movement of infected material. And even the transboundary movement of the potato virus Y, it was genomic assisted tracking, and where the virus isolates, there is a common node between Kenya and US isolates. So the challenges are there is huge amount of data which will be created, analysis and then interpretation and quality control checks and ethical and regulatory considerations, of course, and the fund should be available to do this kind of genomic assisted tracking of global movement. And I have few slides on artificial intelligence, and we are not doing this work, but what is other countries also they just initiated, but there is a possibility artificial intelligence can be used in quarantine and also the emerging and re-emerging diseases. So that basically there are optical sensors which detect the changes in light reflection and absorption to identify the disease symptoms, and thermal sensors monitor the temperature variations which can be caused by stress and infections, and electrochemical sensors, gas sensors, that is volatile organic compounds. Most of the insects are known to produce the volatile organic compounds, and chlorophyll fluorescence sensors measures the stress before visible symptoms appear. So this is, in case of grapevine yellow, like the whole system of artificial intelligence, it has 95.23% accuracy. That is much appreciated. And in tomato diseases also, the slide is not clear, but even in these early blight and mosaic, septoria, blight, and there is 99% correctness was there using this artificial intelligence. And then drones also can be used. Nowadays, I think in India also people are using the drones for insect spraying, but drones assisted plant disease identification using artificial intelligence can also be explored. So both the combinations, but where in quarantine for biosecurity it can be useful is for surveillance and post-entry quarantine inspections. And in Australia, they just started the work in Murdoch University, and they are working with the rat, gecko, and insect, and they're like actually feeding the data and all. It is very nascent stages, but they are working in Australian biosecurity system, and it is not about removing the humans from the process. It is about giving them a more powerful tool that can be speed up and add to their accuracy. Don't mistake that artificial intelligence will do everything and then the human resources are set aside. And in New Zealand biosecurity system also, they are using this artificial intelligence for Polymima. Wherever maize is grown in New Zealand, the digital images are captured and they are trying to improve the system. And even the EU also, they have a smartphone-based citizen science tool for plant disease and insect pest detection, and using for early warning systems. And this, they say, empowers farmers as well as importers border inspections with timely insights for proactive pest management and disease prevention strategies. And there are challenges in artificial intelligence like data availability and quality, and there may be false positives, false negatives, and high implementation cost, and regulatory concerns, whether all these artificial intelligence based detection technologies are accepted by the regulatory agencies like National Plant Protection Organizations, and there may be like need for trained personnel to manage AI tools, and adaptability to climate change is again a challenge. There is a good possibilities for public-private partnership. There is a public-private partnership in our quarantine systems for package houses, post-entry quarantine inspections, but this can be done. It is useful for surveillance and to identify new pests, and but there should be a balanced approach combining AI and human expertise to strengthen the global biosecurity. So this is not my slide. This slide I borrowed from Dr. Lav Kumar from International Institute of Tropical Agriculture, Nigeria. In one of the conferences, it is presented like you can use biosensor or high throughput sequencing, remote sensing drones, artificial intelligence, or multispectral imaging, and then on top of it, everything should be fit for the purpose. And since I am in Tirupati, this movie, everything might have watched, RRR. But for virus diagnostics, it is, the diagnostics should be reliable, repeatable, and reproducible. And capacity building, I said, we have conducted workshops for quarantine and customs officials. And during the last 5 years, we have conducted biosecurity, biosafety training, and more 331 participants we have trained. The beauty of this training programs is we have not only trained the researchers, scientists, and quarantine officials, but also the private industry. And this is my last slide. Like this is my recent visit to International Symposium And Plant Virus Epidemiology. They took us to the citrus growing areas, and then this is one pathogen which is not reported from India, Xylella fastidiosa, and very severe in Brazil. But there is a temptation to bring the material as a reference material, but I have not brought anything. But there are, if I can say in this gathering, Chinese people were collecting a lot of infected fruits to take it back. But I urge you people, if any one of the conferences, if you go, please don't bring any material, even healthy material to I already talked about all these things. There is a need for accredited diagnostic laboratories in India, and there is private industry laboratories are accredited in Philippines, Thailand. I don't know about Vietnam, but in India only private laboratories are so far not accredited, and we need to do that, and we need to have a national plant-based diagnostic network. I will not talk much about it. We recently submitted a project for this national plant-based diagnostic network. This I have been talking for the past 15 years. Only in 2026 we have submitted a project to these are all the pathologists, entomologists, bacteriologists, weed scientists, and even the technical officers both in Delhi and Hyderabad. And I would like to thank Indian Council of Agriculture Research, NBPGR. DBT gives a lot of funding for us, and also United Nations Environment Program, and at the end, IPS Delhi Zone. Thank you. If there are any questions, I can take.
Thank you, Dr. Celia, for elaborate narration about the country's quarantine system. Really, that every conferences you are updating us on these further developments, new developments. Thank you for giving such wonderful presentation. Now, any questions?
Quarantine protocols for the genome-edited crops so far?
Yeah, genome-edited germplasm we have received. The procedure is same. We do the lab inspection, and also the genome-edited, what is the gene, its presence, the protocols is being worked out, like we have digital genomic resources. So anyway, they actually declare what they have done. Once it is declared, developing the protocol to detect that which is there or not. Otherwise, quarantine protocol is safe, and the material will not be grown outside, it will be grown in the containment facility.
Many GM crops received for the something like that.
Can you use the mic?
So many GM crop material has been received. May I know what is the reason, and is for research purpose?
It is for research purpose. India has so far not imported any commercial material for planting. These are all 16,000 samples of wheat or cassava, anything. Yeah, but now the import of transgenic germplasm has come down because of the moratorium. Like many MNCs also have reduced the work on GM work. Yeah.
The protocol may be same, I think.
The protocol is we don't release it to the indenters for the open field. It should be grown in the containment facility as per the DBT guidelines, and we also grow in the containment facility which has high efficiency particulate filter.
Dr. Celia, you made a mention about genome-assisted tracking of lineage. It's a good system, but with the wheat blast, what was your conclusion out of 532 isolates you made a mention? What was the lineage? Where from you?
They said it is Zimbabwe's and also the Bolivia's.
Oh.
Basically, the material has come from Latin American countries only. It is actually related to series of grain imports, wheat grain imports from Latin American countries.
Commercial.
Yeah,
Commercial.
Commercial material. But they have done some genomics also. It is not that they have imported from other countries and they are telling that it has lineage is with Zimbabwe. No, it has come from there.
Dr. Celia, we have been talking about biosecurity zones, and we used to cite other countries like Australia. But still, our system is still porous, and we don't have a monitoring system or kind of biosecurity zone earmark. But what would be the way out? We have been talking about this.
I had been to Indo-Nepal border, that is Raxol in Bihar. People can just walk in and walk out till 9 o'clock in the night. Like Bangladesh and West Bengal, that is the same. The only thing is, initially we thought SARC will do something. But SARC is also not very effective. So, like...
Issue is not that, madam. almost 10, 15 years back, I think we had a biosecurity bill tabled, but not implemented or not passed through.
It has not passed through the parliament. It has lot of efforts were done to make the biosecurity bill, including the that is agricultural biosecurity
Yes,
bill
yes.
in 2013. But that bill was not passed in the parliament.
But,
the reasons we
any-
don't know. Like,
No, reason is any further movement to take it forward, or it is
no, it is
gathering dust.
sleeping. It is sleeping.
I have one comment actually. I am from the very same system. I have no question actually. The paradox here is, in fact, there is a lot of material, and MBPJ has done wonderful work, and all the protocols have been in place, and she spoke about at length about the systems that are in place. But nevertheless, the biosecurity systems of you talk about the New Zealand, Australia, highly advanced. You know, we don't talk about the pilferage also. She talked about bringing pilferage, bringing some samples, pocket and then changing the borders. Those things are not also really problematic. The problem is with respect to the commercial material movement. Any material that is moving out of India is immediately stopped either in the Europe or advanced countries. China also stops a lot of our material. Actually, UK stops even our green chilies. It is not just disease also. Many times a little bit of the pesticide residues, a little bit of, it is all of course geopolitical, situations are also in place. But the biosecurity has to be really, as you rightly said, something has to be done. A lot of, she has also spoken about the impact had the disease not been stopped. She spoke about wheat, I think the soybean and some other crop. Is it
Maize.
maize? So these three crops, NBPJR is doing its job. But a lot of material is coming into the country, and we see a great list presented by her that the recent introduction of many diseases and insects also. So there has to be a mechanism. It's only a comment, as I said. There has to be a mechanism to deal with. Any amount of work done by NBPJR is only to stop the transmission of through seed, through actually germ plasm.
Through germ plasm.
Through germ plasm. So there is a paradox that we need to really understand.
Yeah.
Yeah,
Okay.
recently I have attended a
Thank you.
WTO SPS workshop, and even the younger generation who joined as plant protection officers in DPPGS, they are aware of all these things.
Thank you for all your good presentation. We will continue our discussion further. Thank you. Now I invite Vice Chancellor Madam to give her talk. I think she doesn't need introduction, but still organizers will make a brief introduction about our Honorable Vice Chancellor for her lecture. Thank you.
Good afternoon, everyone. Now I would like to introduce Honorable Vice Chancellor Madam Acharya Enjiranga Agricultural University, Lam Guntur. Dr. R. Sarda Jailakshmi Devi Madam is a distinguished academician and researcher in agricultural sciences, currently serving as a Vice Chancellor of Acharya Enjiranga Agricultural University, Guntur, Andhra Pradesh. She is the first women Vice Chancellor to hold this prestigious position in the university's 60-year history. With over 37 years of dedicated service in research, teaching and extension, she has made contributions in transforming agricultural education and research. She has received D.P. Mishra RN and RN Ponde IPS Best Women Scientist Award from Indian Phytopathological Society, Outstanding Leadership Award in Teaching and Research from Association of Agricultural Technology in Southeast Asia, Thailand, N. Prasad Memorial Award from Indian Society of Mycology and Plant Pathology, Outstanding Women Researcher Award from Springer Nature, Outstanding Agricultural Scientist Award from Dr. B. Vasantraj David Foundation, Chennai. And she holded a position of Professor and University Head in the Department of Plant Pathology, Acharya Enjiranga Agricultural University. And she holded a position of Principal Investigator of RKVY project on production and marketing of oyster mushroom. Now I would like to invite Madam to deliver the keynote address. Thank you.
Good afternoon to you all. The topic selected for the theme of this conference is very vast. Probably many of us know that the plant pathology itself is a vast subject. As of now, the present scenario, many fields are under research in plant pathology. So I may just briefly give about the innovative strategies for sustainable plant health management. So as the topic says, emerging and re-emerging plant pathogens. So as we all already told about the impact of plant pathogens or plant diseases, the emerging diseases are majorly the DNA and RNA viruses of legumes, banana, and Xylella fastidiosa, disease of grapes, quick decline olive, then weed spot blotch, blast, white leaf spot of brassica, rice bacterial panicle blight, potato zebra chip, necrotic ring spot, and re-emerging most of the endemic and epidemic pathogens, then banana fusarium wilt, xanthomonas wilt, wheat rust, potato red blood pathogen, rice BLB phosphate, which is already the major challenge in Andhra Pradesh, sheet blight, foot rot, maize bacterial stalk rot, cassava mosaic, tomato yellow leaf curl, saprophytic fungi Aspergillus penicillium, and bacteria and phytoplasma. So any management of the disease or any pest, it starts with the detection. Once we detect properly or the diagnosis is proper, then everything is solved. So the first, though all these technologies are already the eminent scientist Dr. M.K. Reddy and Dr. Seelachal already mentioned about some of these technologies. So the first and foremost thing is the diagnosis of the plant pathogen. So nowadays, majorly all these technologies are being used for the research. I just briefly mention about some of the technologies. Artificial intelligence, deep learning models with advanced sensing and IoT technologies. These DL and machine learning, which is the brain of modern detection systems, which analyzes the data and then identify the disease. So in this area, we as plant pathologists of Acharya Engineering Agriculture University, we are deficit. We need to get the strength or the expertise in these areas. So I request all the scientists to get trained in these areas. Then CNN, then vision transformers, then advanced sensing technologies where hyperspectral imaging, multispectral imaging, thermal imaging, fluorescence sensing, then IoT edge computing where the sensors and cameras are used for monitoring, then unmanned aerial vehicles or the drones which are used for surveillance, mobile applications, plantics and pheromone, big data analytics, sensors, images which are used for the identification of the disease. So AI and machine learning, so where the 1956 John McCarthy, he has given a man-made thinking power. So these are mainly used in agriculture, especially in plant pathology, because they will be helping in the early and accurate detection of the diseases and pests, optimizing the resource allocation, predicting the disease outbreaks, improving the overall crop management, where they will empower the farmers with data-driven insights, leading to the more sustainable and efficient agricultural practices. So for the complete identification of the pathogen. Then in this image-based disease identification and AI models trained on the crop disease data sets, mobile-based disease diagnosis applications. So here we are having both machine learning and deep learning. In the machine learning, we will use the feature extraction, which will have the color, texture, and the shape features with the final classification. Whereas in the deep learning, both are mixed together and the disease will be identified. Now these are being used. The applications are data collection by using the AI drones, aerial crop survey, disease identification, and creating expert systems. think majority of us are using these mobile applications, Plantix, Plantscope, Agrobase, Agrio, all these they are being used for the identification of the pathogens. So these are the different sets and different models which are used for the different plant disease detections. So now some of these examples are there where already these technologies are being used. Mobile-based deep learning models for banana disease detection, automatic and reliable leaf disease detection using deep learning techniques, mobile-based system for detecting plant disease using deep learning, an application of machine learning technique in forecasting crop disease, predicting rice diseases across diverse agrometeorological conditions using the AI approach. So there are many advantages of these. Accurate, as Dr. Sila was mentioning, measured above 90%, high speed, reliability, works 24/7, can handle complex problems, but some disadvantages are there where we have to minimize the disadvantages also. Then IoT and sensors. So majority of the diseases, they are being used with the sensors, field sensors for temperature and humidity and leaf wetness. You can see the sensors where they are being used for the majorly for the foliar diseases. So weather-based forecasting for rice blast, leaf wetness sensors, grape downy value, sensor-based alerts for lead beta potato. Already we are using in groundnut also all these technologies. Then remote sensing and spectral imaging, satellite, drone, and UAV-based disease monitoring, multispectral and hyperspectral imaging, detection of stress before visible symptoms, drone detection, we can use it for sheath blight in rice, remote sensing for cotton leaf curl disease hotspots, early stress detection of wheat rust affected fields. So all these are there, some of the examples I have given. Several times I have been observing in the agriculture reviews, our Honorable Chief Minister used to tell, you have to spray where the pest or pathogen is present. Don't spray for the entire field. So that is the site application or target application. For that, in our university, we are trying to have the target application by using the hyperspectral image. Then thermal imaging, hyperspectral imaging, and different techniques in the hyperspectral imaging. Then drone-based imagery for precision agriculture. Generally, this started with the military and space use initially, but later, because of the capturing of entire spectrum of color, which has been used in agriculture for the pest or disease identification or detection. Here, the drone imagery, it can assess the soil quality, weed invasion, disease and water needs, and to predict the crop yield or to predict the diseases or even to monitor the crop yield. So these are the different cameras or the drone hyperspectral imaging. So these platforms with the machine learning and image recognition algorithms, they can detect the patterns and anomalies over large areas where we can have the identification or detection of the plant pathogens and also the pests. In addition to all these things, normalized different vegetation indices are also used to assess the plant vigor and stress, which can be correlated with the pest infestations or the pathogen activity. So these can be integrated with the geographic information systems and to support the spatial modeling pest dynamics, where we can minimize the pest uses and also we can reduce the environmental impact. Then next comes the molecular techniques. As already the previous speakers they have already mentioned, just I'll briefly tell about the different molecular techniques that are being used. We know very keenly that these techniques are rapid, highly sensitive, and specific for the identification of the pathogen, both the nucleic acids and proteins, before the visible symptoms appear. So nucleic acid amplification techniques most widely used. PCR, after the invention of the PCR, a lot of changes have come in the plant protection. So in this real-time PCR or quantitative PCR, where we can quantify the pathogen with the fluorescent types, SYBR green or probes, TACMAN, then RT-PCR, then nested PCR, multiplex PCR, and recently the isothermal amplification, where we need not have the PCR machine itself. So loop mediated isothermal amplification, all these things are taken up by us with all our students. Most of the researchers are under the chairmanship of Dr. M.K. Reddy also. So these are the different PCRs, RT-PCR, nested PCR, and multiplex T cell, where we can detect simultaneously the pathogen in the single reaction. Then another area is microarrays, then fish technology, the lab assay in the field condition itself. Then protein-based serological techniques, ELISA, it has been being used in the plant pathology for several years. Then lateral flow immunoassays, which are simple paper-based dipstick tests for rapid onset detection. Then emerging are the fish technologies. Then biosensors, where the biological recognition of the elements, that is the antibodies, are to produce the measurable signal when a pathogen is present. Then flow cytometry, here the flow cytometry, it can identify the pathogen or count the pathogen cells or pores in the liquid stream by measuring the light scattering and fluorescence. Then high throughput sequencing or next generation sequencing, which is proven instrumental to detect the pathogens with precision and speed. So this is ELISA, as we already know. Then lateral flow devices. Then flow cytometry, where the pathogen cells are taken in through the electron detection apparatus in a liquid stream to identify the pathogen. Then recently we have been using this CRISPR technology, clustered regularly interspaced palindromic repeats, CRISPR and associated protein Cas systems used in disease diagnostics. Recently also we have done with the chili leaf curve with Dr. Krishna Reddy sir and Madhavi Reddy madam. So here the ability of the CRISPR-Cas precise targeting of nucleotide sequence has been harnessed to develop the highly sensitive and rapid isothermal diagnostic tools, especially to detect the viruses. So after the discovery of Cas12 and Cas13 endonucleases, they are being used for the diagnosis of the viruses. Then detection based on the transcleavage activity of the CRISPR-Cas12 here. So here the CRISPR-Cas12, it is guided by the single guide RNA, cleaves its target DNA, the generated triplex, that is Cas12A, guide RNA, cleave target DNA. It fragmentizes and surrounding the single-stranded DNA non-specifically, which is known as transcleavage activity. So with this also we can use this technology for the detection of the viruses. Then another way where we can manage or control the diseases is phytomicrobiome. Several times we have been using, still it needs to be emphasized, because nowadays natural farming has already given the focus nationally and also at the state. So we need to focus on the phytomicrobiome, because in the natural farming they are not using the pesticides, they are not using even the fertilizers. Then what is the major role for the health of the pathogen, for the health of the plant, or for the yield that is seen from the natural farming? So here the major role is played by the microbiome, whether it is in the soil or on the surface of the leaf or whatever may be the different areas. So the major role played by the microbiome is helpful for the sustainable agriculture through the natural farming. So this area also, though it is being used, that is biocontrol agents are since age old days, still it needs to be focused, phytomicrobiome. So all these different microorganisms present in of the soil and the leaves, they should be focused to have the antimicrobial or antipathogenic properties. So they offer sustainable solutions for agriculture, plays a key role in plant environmental health and to increase the productivity. Several times all these microbiome, though they may not be registered as biocontrol agents, they can be registered as growth regulators or growth promoters. Several times there is a lot of procedure to register the biocontrol agents, and also it is very expensive. So we can try these microbiome as growth promoting substances or growth promoting agents, so that they can be used for the suppression of the pathogen and also for the growth or increased crop yield. So these are the different rhizosphere, rhizoplane, root endosphere. So these are different roles of microbiome. That is, not only they are against the pathogens, that is biotic, even for the abiotic also they can be useful. There are different mechanisms, we all know, competition, parasitism, all these are the different mechanisms where they can also helpful to make the availability of the microelements, which is not possible normally by the normal process. So the microbiome is helpful not only for the suppression of the pathogens and pests, they are also helpful for the growth of the plants to improve the further yields. Next. So there are several examples. So if you take the rhizosphere, which is majorly the soil zone influenced by the plant root exudates, which is rich in VGBR, mycorrhizae and biocontrol agents, which enhances the nutrient availability, phosphorus, iron and zinc, suppresses soil-borne pathogens and induces the systemic resistance. So there are some examples: trichoderma in rice, sheath blight against the pathogen, then Pseudomonas fluorescens in chili for the wilt and damping of Fusarium and Pythium pathogens, mycorrhizae in millets for improving the nutrient uptake. So several times all these are being isolated from the soil, isolated from the rhizosphere or rhizoplane, but they have to be registered for further commercialization. Then phyllosphere microbiome, which are on the leaves and aerial plant parts, majorly for the epiphytic bacteria and fungi. So phyllosphere bacteria, they can be helpful for the leaf blast and also for the powdery mildew of mango. Then endosphere, that is majorly the endophytic microbes. They are very, very important. Several times these are playing a major role because they are acting as first line of defense against the plant pathogens. And also we take the examples, endophytic fungi in banana can suppress the fusarial wilt, and similarly bacterial blight of cotton can be suppressed by the endophytic bacteria. So it is not enough to isolate the microbiome, whether from the soil or from the plant or from the leaf, but how best we can engineer the microbiome is the present scenario. So microbiome engineering is very, very important. In addition to that, consortia of the microbiome is also very, very important. So manipulation of microbial communities for desired traits is the present day research. So use of multi-strain microbial consortia, synergistic effects on the disease suppression and also on the growth, where they can be performed well compared to the single microbe. Then for the management of the pathogen, there are recent techniques, decision support systems, which are data-driven disease prediction models, advisory systems for fungicide application timing, crop and location-specific recommendations. So these technologies can be utilized to use them in the management of the pathogens. Then another area of research which is used for the management of the pathogens is nanotechnology. Though it is being taken up vastly in TNAU and also at ANGRO, but it is to be further studied for the betterment of the nano products. So nanopesticides, fungicides are nano delivery systems for targeted nutrient or gene delivery, advanced diagnostic, metal nanoparticles, silver, copper, zinc oxide for direct antimicrobial action. So all these silver, copper and zinc oxide nanomaterials have done by using the chemical extraction and also by using the trichoderma synthesis. Then nanobiofungicides, effective alternate to the traditional fungicides, nanocarriers for solubility, stability and targeted delivery, nanofertilizers to boost plant health. Nowadays IFCO and Coromandel, they are going for the nano urea and nano DAP. So inducing plant immunity, nanoparticles trigger plant's own defense system, making resilient to the infection. So major benefits are we can reduce the chemical usage, environmental friendliness, improving the crop yield, fresh and warming. So these are the nanoparticles where we use during our research. So utilization of nanotechnology integrated with the bioagents, nanoparticles synthesized by using the microbial biocontrol agents. These formulations can be highly helpful because of the high surface area volume ratio, improved effectiveness, less toxicity, higher solubility, durability, versatility. All these are the benefits, but still we need to have this toxicology studies. When once we develop nano products, they have to be tested for the toxicology for further use or for further registration. So these another area which is now being focused is biochar. Biochar is also very, very important, majorly for the soil-borne pathogen control. So we used it for the groundnut stem rot disease. So harnessing biochar for sustainable management of groundnut stem rot for the soil health management. So now for any crop we can use or recommend this biochar. Even our government also focusing on having these startups on this biochar. Then genomic and molecular insights for plant disease resistance and resilience. That is, after doing all these things, when once the variety with the tolerance or variety with the resistance or any material, planting material with the resistance, if it is given, it will be more helpful to the farmer. So there are many techniques. So genomics, where genome-wide association studies, they are used to pinpoint disease-resistant genes in the germplasm. Pangenomics to identify the novel resistance in the wild relatives. Then I have been telling that most of these technologies are being used already. RNA technology or RNA silencing. So to create disease-resistant crops by engineering the plants to produce the silencing RNAs, which are offering the target protection against the viruses, fungi and pests. So here in this procedure, specific genes are turned off or silenced by targeting their messenger RNA or degradation, preventing the protein production as an antiviral defense. So this method is also used for the disease resistance. Then the expression of virus-derived sense or antisense RNA in the transgenic plants conferring RNA-mediated virus resistance seems to induce a post-transcriptional gene silencing. Then other areas are fetal mapping and functional genomics. Most of the times now we are focusing on these transcriptomics, proteomics, metabolomics. So transcriptomics, they analyze the gene expression changes during the pathogen infection, where we will be studying the host pathogen interaction. Then proteomics and metabolomics, they identify the proteins and metabolites involved in the disease resistance while doing the breeding. So advanced breeding approaches. Nowadays we are also using, even Bhaskaran and others are using this technology for developing the resistant varieties. Marker-assisted selection, genomic selection, DNA markers to identify the desirable genes. Then another area is genome editing, where the CRISPR-Cas also being used. So CRISPR-Cas based genome editing for precise trait improvement, development of disease-resistant climate-resilient varieties, enhancement of the yield, nutrition and also tolerance to distress. So this is a process where genome editing is being taken place by using the CRISPR-Cas9 and 12A. So CRISPR-Cas locals and the CRISPR-Cas immunity steps. The applications of these technologies are at the molecular level. We can have the gene deletion or gene insertion or gene replacement, then base editing, transcription modulation, DNA labeling, etc. Then for the practical applications, biological research, human medicine and biotechnology, and also in agriculture. Whereas in agriculture, for improving the yield, for improving the pest and disease resistance, herbicide tolerance, improved nutritional quality, in all these areas we are using these genetic technologies. These are the different applications for crop improvement by using the gene editing, genome editing. By using all these, still we have some certain concerns. Regulatory and ethical considerations. In India, we have SDN and SDN2 genome edited crops, which are exempted from the GM regulations because they are not inserting any gene, they are not deleting any gene. That simply they are modifying for the rest. That's why they are not under the genome regulation, the genetically modified regulations. Then biosafety and food safety. Biosafety, just now we have Sheila talked about the biosecurity. So biosafety, food safety and environmental concerns are another area. The need for transparent policies and public awareness. Ethical use and equitable access for the farmers, all the farmers equally, that is also majorly essential. Then next comes the role of industry. Though we as government agencies whether it is ICR or SAUs, we are doing lot of research, but we are not going up to commercial level because the industry plays a major role at the point of commercialization. So in the crop protection and agrochemicals, the industries they have to play a major role to produce the less toxic, lower dosage, more specialized chemicals, and also in the agri-biologicals and sustainable solutions, they have to give the sustainable solutions to have the bio inputs, biopesticides, biostimulants. Then in the area of biotechnology and seed science, pest and disease resistant varieties. Then plant tissue culture industry, virus index, superior quality planting material. Then stewardship and farmer education, where industry also has to play a major role in training the farmers and retailers to promote the IPM and also judicious use of the pesticides. Then digital and precision agriculture, AI and data analytics for crop profiling, pest surveillance, precise farming solutions, and in some of the areas there is a separate area where horticulture or plantation crops, specific support should be given by the industry for the specific crops to provide high quality disease-free planting materials and technologies. Then startup driven innovations, though there are startups, but they are not being commercialized. Unless it is being helped by the industries, the innovations may not be fruitful. So startup driven innovation, market oriented solutions, rapid diagnostic kits for early detection, and scaling the research outputs through the commercialization. And latter, we together, we have to strengthen the linkages among the researchers, farmers and the market. Then stakeholder linkages, research, industry, farmer continuum, that is whether it is ICR, SAUs, all we the scientists with the agri-industry and the farmers, we need to coordinate, we have to cooperate and coordinate for the transfer of the technology. Then role of FPOs, nowadays FPOs are also playing a major role. So role of FPOs and extension agencies, FPOs should be as hubs for the input supply, for the advisories and for the market intelligence, because last year we have seen all the papaya, onion, mango, all these, though there is a lot of produce from the farm, but there is no market. So this is the challenge. So we need to think about the market linkages also, and all the extension agencies, that is KBKs and other extension agencies, they should enable the field level dissemination. Then next comes the public-private partnerships. So we need to have the collaborative research and development in the seeds, bio inputs and diagnostics, scaling innovations through the government and industry synergy. So I thank the organizers for giving the opportunity. Though the subject is vast, whatever area we have expertise, we need to go ahead with all the updated knowledge. Thank you.
Thank you, madam, for your bird eye view of plant pathology, current area of work, and also for the future perspectives. Do you have any specific questions? Madam has covered almost the entire area, starting from traditional work to the modern technology, including genome editing, and that way she has covered the entire area, and including nanotechnology, AI, and all the modern diagnostic tools involved in biotechnological work that are applicable in plant pathology. Most thank you, madam, for giving such elaborate work. I think since there is no further discussion, this session we will close the inaugural session along with the keynote speeches. So we had three speakers, eminent speakers, Dr. Krishna Reddy, Dr. Celia Charam, Dr. Honorable Vice Chancellor. They have covered diverse areas, but almost it was like a classroom teaching. I learned again this refreshed on this seed virus transmission and also the quarantine regulations and other. The third paper, Mrs. Madam has given a complete overview of what we should project for the future of plant pathology research. And I thank all the three eminent speakers for sticking to time, and we were able to complete these all the three keynote lectures in time. And over to organizers for further announcement. Thank you, thank you. Thank you all the speakers.
Thank you, sir. Now we'll break for lunch, madam. So lunch is arranged beside the auditorium. Please, all the delegates and the participants here, please join for lunch. And we will again meet for theme 1. Theme 1 session, it starts at 2:30 p.m. 2:30 p.m. All the oral presenters, please be attentive and come by 2:30 p.m. here. Lunch is arranged beside the auditorium. Just please join all the delegates. Please join the lunch. Thank you.
I request all the participants, please come inside. Auditorium, please. Session will be starts very soon. The first speaker is Dr. M.K. Naik sir. The topic is key challenges in plant pathology in the next decade. Before that, I will read out the biodata of Dr. M.K. Naik sir. It is my privilege to address the biodata of Dr. M.K. Naik sir. And Dr. M.K. Naik sir is distinguished agricultural scientist and academic administrator with more than 3 decades of service in Konali, Kumta, Uttara Kannada district, Karnataka, and also he has risen from a humble background to become one of the most respected plant pathologists in India. Dr. Naik sir completed his BSc Agri with Roll of Honor from the University of Agricultural Sciences, Bangalore, followed by MSc Agriculture with multiple Best Outgoing Students Awards and ICAR JRF at US, Bangalore. Sir secured second rank in All India ICAR JRF examination and obtained his PhD from IRA, New Delhi. He also completed a postdoctoral fellowship at ICRISAT, Hyderabad, and holds a PG Diploma in Human Resource Management from IGNO. He passed the ARS examination with second rank at the All India level. And Sir was specialized in plant pathology. His research spans biodiversity of plants, fungi, integrated pest management, mycotoxins, biological control, PGPR, and bio-entrepreneurship and sustainable agriculture. His scientific contribution include the identification of antibiotic producing and climate resilient microbial isolates and development of IBM modules, discovery of new pathogen variants, microbial resource deposition in international gene banks, and release of improved crop varieties. He has also been instrumental in establishing biocontrol laboratories and promoting bioinputs at the farmers level. Dr. Naik sir has published over 200 peer-reviewed research papers, authored 15 books, contributed more than 70 book chapters, delivered numerous keynote and lead lectures, and actively participated in national and international conferences. He holds 3 patents and has significantly contributed to agricultural education, research and dissemination, and farmer outreach through TV and air programs also. His professional career includes serving as scientist and senior scientist at ICAR, IIHR, associate professor and professor of plant pathology at US Harvard and Raichur. Dean PG Studies, Director of Research, Director of Education, Director of Extension and Vice Chancellor of the University of Agriculture and Horticultural Sciences, Shivamogga, from 2018 to 2022. As Vice Chancellor, he played a pivotal role in developing UHS as an integrated farm university, establishing infrastructure, research stations, incubation centers, and promoted innovation, entrepreneurship, and sustainability. Under his leadership, several centers of excellence, model farms, seed units, post-harvest centers, apiculture units, and dairy units were established. He successfully monitored and implemented hundreds of externally funded research development projects amounting to several thousand crores, contributing significantly to farmer income enhancement and rural development. Dr. Naik sir is a recipient of numerous prestigious awards including ICAR, Bharat Ratna, Subramaniam Outstanding Teacher Award, and also Fulbright Nehru Fellowship, ISCA Young Scientist Award, Karnataka State Young Scientist Award. University Best Teacher Award, multiple Lifetime Achievement Awards, and National Memorial Lecture Honors. An accomplishment teacher, researcher, administrator par excellency, Dr. M.K. Nayak sir continues to inspire the agriculture fraternity through his vision for excellence in education, research extension and human resource development. Thank you, sir. With this introduction, I invite Dr. M.K. Nayak sir to deliver key challenges. Theme of topic is key challenges in plant pathology in the next decade. Thank you, sir.
Thank you, Professor, for those kind words. Honorable Vice Chancellor of this prestigious university, Angru, Dr. Sharda Jayalakshmi Devi, all the distinguished delegates, ladies and gentlemen. The stage is set for very sumptuous, delicious, tasty food, and the AC is put on. Now the challenge is for the speaker, whether to keep them, whether they will sleep or not. Challenge is for the speaker in a lighter vein. I will try my level best. I did not speak in the morning thinking that I will be taking those 10 minutes here. That is the reason I did not speak now. Madam will permit me. Can you put this light off? I have taken the topic on key challenges in plant pathology in the next decade. If you look at the demographical changes taking place in the world, we have seen that the first billion, or what we call 100 crore people appeared on this planet back in 1800. That means we might have taken thousands of years. And the next billion came in 1930, 200 crore in 130 years. And the third billion came in another 30 years. And the next 200 billion, 500 crore came in 1987. And last 2010 we had 7 billion population. And this will continue to grow. Probably we will be 930, that means 9.3 billion crore by 2050. And as far as India is concerned, probably we have surpassed China on 25th April 2025. And this will continue up to 2070, the growth rate. But there is a bigger role for plant pathologists because the major crops, losses caused by, that means as much as 40% yield loss, which will be around $220 billion, approximately around 200 lakh crores. That is a very big money, including the major crops. But as far as India is concerned, in agriculture, we have traveled a very 80 years of very significant and incredible journey. A journey in which we have produced food production of 6 times higher, 352 million tons. Horticulture 12 times, again around 340 million tons. We are number one perhaps in milk production, almost about 200 million tons. Most importantly, the morning we have had a good breakfast. Afternoon, madam was kind enough to provide the food. Evening, I am sure we will have a dinner also. That means 140 people, morning, evening and afternoon. Together 400 crore meals every day. Probably nowhere in the world you will find a country feeding 400 crore people every day. That is the biggest achievement of independent India. Although we say nearly 400 satellites have been launched by India, yet I feel this is the biggest reason to celebrate for independent India than satellites, with all respect to the space scientists. Okay? Then, also it is important to note that India is the only country in the world where we have enacted Food Security Act, promising that we will be able to feed 80 crore people under the public distribution system. That is also one of the biggest achievement. But today, all this was possible because of three ingredients. That's what we call as, political will, farmer skill and scientific skill. Together, this has made it possible. But at the same time, we should remember that we are not as happy as we should have been, because the Human Development Index is 130 out of 189 countries. Our Global Hunger Index is 102 out of 119 countries, and some of those countries like Bangladesh and lesser known are ahead of us. That is also not a good statistic. More importantly, hunger and malnutrition is still persisting. This beautiful picture is not a beautiful picture, it's a picture covered by Pulitzer Prize award winner, which is as good as Nobel Prize. So that itself speaks, the world is, not everything is smooth. A hungry person has neither religion nor reason to ask. He will only ask for food. That's why I think we cannot build a developed and strong India on the shoulders of stunted children or dwarfed children. Next one. So there has been a very unprecedented changes, transformational changes which has taken place. We were rather deficient, now we are sufficient. We were importing, now we are exporting. We are waiting for food security, but that we now want nutritional security. We were talking about green revolution, but that now we are, since morning you have seen people have been talking about gene revolution, genome editing. That means one gene of BT has changed the textile industry shape, and we were importing that we are exporting now. Similarly, we were from subsistence agriculture to commercial agriculture. From chemical farming, we are moving towards biological farming. So that is the state of affairs. So, but challenges of Indian agriculture are plenty. There is a rising rural distress. We have been shouting the slogan of doubling the farmer's income, which is not yet realized. Shrinking farm size. In the process of green revolution, we have left a lot of water footprint and carbon footprint. And there is a climate change is haunting us. With all this, probably we have plenty. Please go back. The income, if you ask today, the monthly income of a farmer is, average is 10,000 around. That means 1,20,000 average. State like Orissa is 5,000, and there is a state which is going 22,000 also. But the point is, it has not been realized. The MS Swaminathan report says we have to have a minimum of 2.5 lakhs for a family of 4 people in a hectare field. And that was back in 2004. And in 2025, the inflation rate and everything, it should be more. So that is not realized. Next one, please. So, in other words, the so-called food security is incomplete in the absence of biosecurity. Therefore, just a tiny microorganism like COVID virus has shaken the earth. It has locked down the globe, disrupted the supply chain, and changed the production system, and has influenced the vulnerable sections of the society, including developing countries like India. Therefore, we need to attend the biosecurity, which is a must for food security. Based on this, American Phytopathological Society conducted a survey last year with 3 questions in mind. The number 1 question is, how do we prepare the challenges of plant disease to meet the food security and nutritional security? That was number 1. Number 2, where the field of plant pathology will be heading for, where we will be in the next decade. And the third question was, what was the advancement needed for the next 10 years to meet the challenges? These were the questions administered for 661 survey. Let's go back. 661 survey, as many as 39% of the faculty and 39% of research scientists, postdoctorals and graduating students. And then the results. These are the key challenges listed, as many as a dozen of challenges. One is that we are emerging into one health or one world. Prime Minister often repeats this. Climate change, that's what Atherton wrote it as disastrous plant pathology, a branch of disastrous plant pathology because of the climate change. Crop resistant gene identification and cloning. I'm not only telling the challenge, the opportunities as well. Antimicrobial resistance, food safety including mycotoxin, accurate detection, that is the Clean Pan program what Dr. Krishna Reddy is also looking at, and emerging pathogens and toolkit. Next one. The challenges listed, again, biotech, CRISPR gene editing, omic application like genomic, proteomics and metabolomic approaches, artificial intelligence, madam has elaborated, my job is reduced here, and machine learning, and one of the most challenging pathogens are phytoplasma, wall-less prokaryotes, and then they are emerging insect transmitted plant pathogens, vectors, susceptibility genes and the biocontrol. We are still, although we have been talking about biocontrol, it has hardly covered 9% of the area, leaving the 91% of the area uncovered. IPM is still a distant dream. Entrepreneurship is entering into plant pathology. Next one, please. I just saw the literature also. The APS also invited all the authors world over to contribute on these particular issues. And there are so many chapters on climate change by Miller, again in 2000, for the information of PG students if they are here. This is the literature you get on Miller, then disaster plant pathology by Atherton, including the digital solution, multiple challenging pathogens by Higgins, and pathogenicity and resistance mechanism by Van. These are some of the issues, even the available literature says. Next one. And there is a chapters available on antibiotics, biocontrol, RNA, biofungicides and bacteriophages. Very, very eloquent presentation is available in APS last year. Next one, please. So as is the first one is application of one word. So we see probably crop health, animal health, human health, fish health, and including the poultry health. All these are inseparable. That's what we call it as concept of one health. You can also see, although the concept was conceived back in 1964, that is why July 6th is celebrated as World June's Day, Zoonosis Day. And today, food safety, antimicrobial resistance, ecosystem, beneficial and foodborne diseases, these three are listed, are the topics of relevance for One Health. Next one. So why should we secure a holistic health? I think we have forgotten that human health is dependent upon every facet, plants, microbes, birds, fish, wild animals. Whereas plant health is actually essential for the survival of all the forms of life. That's why I think the Prime Minister's office has initiated one of the One Health issue recently, just 2 months back, and they have earmarked several crores for One Health issue. I would say that the plant health should find a priority, because in the absence of plant health, probably human being cannot survive. Next one, please. I have taken this slide just to say how important it is to have plant health and human health. Look at here, during the COVID era, we listened during the second wave, we heard so much about black fungus, white fungus and yellow fungus. I still remember when I was in Shumaga as a Vice Chancellor, one of our employee underwent surgery because of the mucor infection. His eyes were, he underwent surgery, and it was looking so awry that it was black color. The color is black, that is why the skin becomes so black. It's called black fungus. And the other one is white fungus, where it is a superficial infection on the tongue, genital organs, even on the surface. But systemically, but the most important is aspergillosis. Intensive care unit. My daughter is sitting here, she may be aware of it, that doctors will be treating the intensive care unit, but all these fungi are a very good source in the intensive care unit, and the pneumonia, everything is affected there. That's why it is important, if you go to the next slide, the same fungi also infect crops. So it is all those fungi, aspergillus, penicillium, etc., they infect immunocompromised patient. Here also, it is found in those immunocompromised crop. I say immunocompromised because if it is a red chili, you get infection. If it is a green chili, you don't find. Green banana you find, but in red banana you find it. That's because they have lost their immunity. Same is the phenomena even in the case of human beings. So, but they are so similar, but they do infect both of them. Otherwise, they are very, very weak parasites on plants. But they are very strong on human being, because on plants we spray fungicides, but we cannot spray fungicides on human being. That's why any infection on the lung is going to be a tough job for any doctor to treat. Next one, please. Most importantly, the food safety issue. Today, Food and Agriculture Organization of the United Nations has declared 1/3 of the people, commodities, food commodities are infected by foodborne diseases. India, there are as much as 75 million illness because of foodborne diseases. And last year, the total data says 3,25,000 hospitalizations and as much as 1,800 deaths. So with this, probably it has become a health hazard, international trade issue, and particularly in the modern era, when you say ready to eat food, probably we have to be careful whether we invite a foodborne disease. Therefore, the pesticide residue, antibiotic residue, all are in the food safety issues then. AMR is now a global phenomenon. We have been conducting lot of seminars on this, because the antibiotic resistance, particularly it is a medical issue, social issue, as well as economic cost issue. There are as many as 2.8 million illness and 35,000 deaths reported because of antibiotic resistance. And the global death is reported to be about 10 billion. That means in the economic cost, if you estimate, it will be $1 trillion. This is the biggest challenge, because we see as plant pathologists, we are also culprits, because during our days we have asked them to spray with streptocycline, tetracycline, every cycline. And that ended up because even pharmaceutical industry, even aquarium, all they were applying antibiotics that has entered into the food chain. Even a small fever today is not easily curable by an antibiotics. Next one, please. So this is how you can see here. Poultry birds, as a prophylactic agent, we are administering antibiotic. We also use in the cattle feed. We also give for piggery, aquarium, prawn culture. I think Andhra Pradesh coast is well known for prawn culture, where a lot of these antibiotics are administered. As a result, they have become easily resistant. Today we don't find, I think penicillin is one of those. Back in 1927, Alexander Fleming discovered. We are yet to celebrate the 2027 as the centenary year of discovery of penicillin. But today, although it is a broad spectrum antibiotic, but it is now offering resistance. Next one, please. Added to this, I have just given one example. Last time ADG asked me whether you have a data on resistance for bacteria in plants. So this is a bacterial rot of cruciferous crop caused by Xanthomonas campestris, where we find multiple genes offering resistance. As many as nearly 60 isolates found one gene resistance. There were some more isolates found double gene resistance and triple gene resistance also. This is where I think your streptocycline cannot be used, but we use very frequently on bacterial blight of rice, even on bacterial black rot of crucifers. Next one, please. Similarly, fungicidal, there are plenty of evidences. I am sure Anguru must have also listed, including blast disease, triozoles. There is a resistance. Especially for oomycetes fungi, whatever we are spraying, metalaxyl, and even Phytophthora mealy, late blight fungus, they are all resistant to metalaxyl. And then Colletotrichum recently in Shimoga, there is a big epidemic of leaf spot and anthracnose of arecanut. And government of Karnataka decided to give 4,000 rupees for every farmer for spraying hexaconazole, but that is not at all effective because the Colletotrichum has got resistance now. Similarly, this succinate dehydrogenase inhibitors, isophetamine, pideflumetol, all these, even DMI, tebuconazole, all these have developed resistance. I think in another next 10 years, fungicide application, because we have a habit of single bullet approach. Chemical fungicides, we are very happy to have it. But the thing is, they have already developed resistance. Definitely, we need a management. Last week, the government of India has enacted a pesticide management bill. That is going to be very important, crucial for management of resistance. Next one, please. Just an experiment that we took up in Raichu, one of my PhD students in collaboration with Baba Atomic Research Center. Carbendazim tolerant trichoderma, because trichoderma could not be a panacea for everything. So trichoderma was, whenever you go for integrated pest management, you will be using fungicides. That's why we wanted to know how best we can develop a tolerant one, carbendazim tolerant mutant by utilizing gamma rays and induced mutation. We could find the double mutant as well as the single mutant here, with the concentration of the wild mutant was having just 0.1 effective concentration, whereas the double mutant and the single mutant 1,000-fold increase of tolerance of fungicide. Similarly, when we go for beta tubulin profiling, we could find the wild mutant and this one, when the homology was noticed, we could find far of this difference among the two isolates of mutants. That's what we want to say, that there is possibility of inducing mutant which can be utilized in our integrated pest management program. Next one. Next one, please. As I already told you, climate, probably the cyclonic, frequent cyclones that we are facing world over, the torrential rain, the frequent droughts, all have impacted. It's very difficult at this stage. Madam, probably during her inaugural address, Honorable Vice Chancellor also mentioned 1845 Irish famine. And the entire decade was a hungry decade for the island people, mainly because the previous 1845 6 weeks had a low temperature of 1.5 to 6 degrees. Nobody has assessed that, because that time there was no assessment of climate. Today we have a microclimate assessment, but that's what it says. The whole Ireland, which had a devastation mainly because of the climatic changes. Potato blight of 1918 was influenced by World War I, because during the World War I, copper was not released by Britishers, and there was no copper sulfate usable. As a result, that ended up, they have to lose their crop. Blight was so severe. So there is a saying, for want of a nail, the shoe was lost. For want of a shoe, the horse was lost. For want of a horse, the battle was lost. And the Britishers lost the war because of the copper. So that is the story. I think southern corn leaf blight is another beautiful example of where America used cytoplasmic male sterile line all over Miami to Maine. So 70% of the maize was covered by cytoplasmic male sterile line. The mistake made by Americans was that they put all eggs in one basket of cytoplasmic male sterile line, And when the basket fell down, the T rays, it captured, and the entire cytoplasmic male sternal line was washed out. As a result, they lost countries 30% grains. And America by 1970 were very developed, so they did not suffer. But had it been in India, we would have suffered. Next one, please. So these are some of the minor diseases in rice. I am only giving an example. Bacterial leaf blight was a minor disease, but now it is a more aggressive. Tungro virus, which was very sporadic here and there, now you find in Uttar Pradesh as well as in this Coromandel region. Sheath blight was localized in hills, but now you find in plains as well. I just wanted to give an example where we have worked on arecanut. Arecanut is a lifeline for farmers of Karnataka, not only Karnataka, for India as well. 5 lakh hectares, 10 lakh tons, but at the same time, because of the climate change, we could find minor diseases. I remember during 1983, I did my MSc thesis on Coletotrichum, beetle vine. Today, you cannot believe it, this beetle vine, this anthracnose of arecanut entered Vidhan Soud also. And they called me to give what are the measures. So that is the amount of, at every farm, it was estimated a loss of 225 crore. So that means the minor disease has become major, and those which are almost back-seated have come to the front, and that is why now arecanut industry is in danger. So this is the training we conducted because we were not finding the people for climbing and spraying. So this is you can imagine the urgency, because drone was not helpful. So that's why I just wanted to say the kind of urgency we have. Next one. So if you look at the 140 years data, just macro data, we can find between 1960 to 2000, or between 1940 to 1990, the difference is 0.4 degree before that, and then after 2000 you find again plus 0.4 degrees Celsius. So this is the biggest change of 1 century. If you look at the past 1000 years data, northern hemisphere, we would find the major difference in the last 1960 to 2000. 40 years data, you see how the temperature is shooting up. We are responsible because the kind of lifestyle that we are using, flights, isn't it? Or your refrigerator, air conditioner, whether it is required or not, all that has contributed for climate change. Next one, please. A beautiful example, probably Madam also mentioned Paxinia Park. This is purely an example of climate change. Of course, we are happy. In 1960s, it was a very peak epidemic. Today, we are able to manage this actually because of the vertical resistant varieties deployed across the epidemic zones. That's why what I wanted to highlight here, whenever there was a cyclone in the Bay of Bengal, the last 20 years data says, whenever there was a Bay of Bengal cyclone, the incidence of rust was high. Whenever there was no cyclone, then there was no rust. So this is directly correlated in the Bay of Bengal cyclone associated in the Andhra coast. Next one, please. Morning, Dr. Celia and Dr. Krishna, they also mentioned so much about lineage and migration. I just wanted to say that UG 99, Paxinia graminis triticale, moved from Kenya to Ethiopia, Yemen to Sudan. These are all climatic influences. Maybe locust movement in deserts is also one of the outstanding example of climate movement. But definitely, we were all waiting to greet the UG 99 in India, and we were preparing. Fortunately, next one please, we did not get the UG 99 in India. Maybe when we prepare well for the examination, question paper will be easy. Similar to that, the CIMIT, where we went and conducted trials and all the varieties screened, we were able to manage. Today, we are predicting that it will reach China, but it has skipped India right now, waiting for that it might enter into China. All these are the climatic influences at the global level. Next one, please. I don't want to touch upon this because we are more interested in variety of life, okay? Increasing tourism, cut flowers, introduce pasture, wood, everything is contributing for our migration. Along with that, we move the planting materials. Along with the planting materials, we meet the pathogens also. I have a long list of pathogens intercepted, which I am not going to display here. If you look at the kind of rejections, aflatoxin, mycotoxin, and then some of the pesticides are the major ones which were also used for rejecting our consignment whenever we import. But point is, the pathogen interception is not as easy as what you do with pesticides or with your mycotoxin. But the pathogens, generally what Dr. Sharad Babu might say that germplasm exchange you are doing it. But when it comes to commercial, perhaps if you don't employ, if you don't implement the National Biosecurity Act, so sooner or later we will be ending up in more of problem. Next one, please. So this is the one like I already explained. Next one, please. This is another, I just wanted to say this is an issue of disease resistance, potential variability of most pathogens. That means the battle between the pathogen and the breeder will continue. Continue till when? It is a never-ending process. That's why breeding for disease resistance is not a procedure to stop anywhere. As long as you get variability, you will continue to do that. Next one, please. I have taken one example of wilt of pigeon pea. In fact, back in 1980s, there was one variety called ICP 8863 which occupied 1 million hectares in southern India, including Tamil Nadu, Andhra Pradesh. In Karnataka, we called it in the name of Maruti, but it is a resistant variety, ICP 8863. But that broke its resistance, and that's why I gave this problem to one of my students. Next one, please. He worked in ICRSAT using the pointer. So this host differentials, utilizing this host differentials, isolates collected from New Delhi, Uttar Pradesh, Madhya Pradesh, Maharashtra, Telangana, Karnataka. We could make a profiling of the virulent isolates. Next one. And as many as pathotype 0, pathotype 1, pathotype 2, pathotype 3, they were added to the existing flora. This only indicates that you need to deploy the gene resistant to these pathotypes suitable for that particular location. So that is the only meaning of that one. Next one, please. This is another variability is also seen. Chemotypic diversity is visible among the mycotoxin fungi. This is the one project we operated. I just wanted to say in groundnut and chili, there are isolates which are showed zero toxin. That means they are non-toxigenic. But there are isolates showing around 2,200 microgram. That means any of your consignment is about 10 microgram, that is rejected. So the possibility of evolution of pathotype or chemotype in the existing ecosystem is highlighted here. Next one, please. So we actually contributed for global assessment of chemotypic diversity. Isolates from America, Africa, Argentina, Latin America, including India. From India we participated. We could notice that generally, I think students would know, when you say Aspergillus fungus, parasexuality is the cause of variability. But here we could notice it is not the parasexuality discovered by Ponte Corvo. It is the sexuality which is leading for chemotypic diversity. That's why we get so much of diversity in production of toxin. Next one, please. Biosafety issue. I think if you look at the last 2-3 years data, you will find, including probably Guntur chili, isn't it? There are 2 issues for which our chili is rejected from outside India. One is for pesticide residue, the second is mycotoxin contamination. Almost 24% of our export is rejected. Next one, please. So they are entered into the food chain, B1, B2, G1, G2, these are the derivatives, and they are very ubiquitous, and they are very weak parasites, but they elaborate the toxin when it is consumed. So the history goes back to 1860, where it was called as turkey disease, where the chicken gets dwarfed when they eat the contaminated groundnut meal. Next one. So you can see whenever there is a contamination of aflatoxin, you would find the animal skin getting scaled, the chicken getting dwarfed, and the liver gets bloated. So you find both acute aflatoxicosis and chronic, long duration if the aflatoxin is accumulated, that also ends up in liver cirrhosis. Next one. So these are the types of aflatoxin with chemical structure. Next one. So what is totally summing up this one, what we need is a quick detection on site, right in the, whether it is in the farmer's field or harvest yard or market yard or storage or consumer's point. In the food supply chain, there should be an on-site application of, deployment of tools, very quick detection, and then it should be very cheap. But we don't have a cheaper ones. We have techniques like HPLC, ELISA and all those, but they are all sophisticated, only laboratory confined. But what we need is one that can be applied by a KVK scientist. Next one, please. So this is one simple technique where you find there is a ring formation. That is the indication. This is coconut meal agar. Indication of presence of toxigenic isolate. Next one, please. So these are, of course, even biosynthetic genes identified, NORA genes, WER genes. Next one. One of the management practices that, because you can't go for pesticide spray for groundnut or chili, the best way is using microbiome. Madam has also mentioned this. So we have some of the best fluorescent Pseudomonas isolates having antibiotic producing capacity, as well as sideropore production, hydrogen cyanide. These are the wonderful quality of a biopesticide which can be used and we use for. Next one, please. So when we use this as a pre-harvest rate before 10 days of harvesting, we could find by Pseudomonas putida, we can reduce up to 70% of infection. You can see the middle ones having no infection, whereas the control is having sporulation. That is the indication. If it can inhibit the sporulation, that is a wonderful biopesticide. Next one, please. Then we conducted IPM modules almost in 4 districts of Hyderabad, Karnataka, what we now call it as Kalyana Karnataka. But Raichur, Koppal, Gulbarga, all these regions adjacent to Hyderabad, we could find that model could only end up in aflatoxin of less than 2.84 or average of 7.58. That's a good indication that you can export your chili. Next one, please. I just move on to, we have been talking that we should not do plant pathology in isolation. Today we are moving towards bioeconomy. Professor Abdul Kalam said, knowledge, when you put into action, should help in reaping the wealth. Knowledge, action and wealth. Today we have all the knowledge available. We have plenty of biological resources, plenty of biological solutions, and plenty of biological processes. But end product is not as much as we expect. But the country is moving towards bioeconomy. One of the renewable biological resources is microbes. Next one, please. So microbes, very myriad living being, not visible, but they have their output is $1 billion question. Some of them, I think Reddy Lab can tell you. You have a Reddy Lab in Hyderabad, isn't it? Vitamins, protein, they are selling it. Like that, microbes can biotransform. Yeast is a single cell protein. Useful metabolites are produced, enzyme activity, biological control, cheese formation, food yeast, antibiotics. All these are various industries, entrepreneurship, which plant pathologists can think of. All those young plant pathologists sitting on the backside have to come to the front to take up the entrepreneurship. Then only it is possible to take up the bioeconomy. Next one, please. As you can see, by 2022, these are the various segments of bioeconomy, including biopharma, COVID vaccine, bioagri, including the BT cotton, biopesticide, enzymes, biofuel. Together, almost $150 billion country has achieved. This is the real achievement. and we have achieved it 2 years ahead. We are supposed to achieve it by 2025, but the country has achieved it in 2023. As a result, the cabinet has now gone for, next one please. Yeah, cabinet has gone for a revised target of $330 billion by 2030, which is also possible. I think unless the bioagents, biofuel, biofertilizers, BT vaccine, all these put together, apart from your COVID vaccine, these are all left to the medical scientists, but that can be enhanced by entrepreneurship. Next one please. These are all the possibilities. Waste to wealth, diagnostics, vaccines, biofood, biofuel, pesticide degrading microbes, possibilities. Next one please. That's why now pesticide residue degrading microbes have been also identified. There are plenty, even radioactive waste, oil spillage, 2,4-D weedicide, chlorobenzene, all these are biodegradable. There are plenty of microorganisms identified and available in NBAIM, National Bureau of Agriculturally Important Microorganisms. Just next one please. So these are some of those. Yeast is probably able to degrade DDT. Proteus vulgaris can degrade DDT. Aspergillus niger, aldrin, penicillium can degrade malathion. Trichoderma, even malathion, mucor can also degrade. These are the isolates available. Next one. I have just one example from our own laboratory. One of my students, we have patented one product process that is a microorganism azotobacter, where it can degrade up to 95% of pendimethalin, chlorophyrin% also 90%, carbendazim up to 90%. But we got the patent, but afterwards I moved from Raichur. It is still in the isolate. But that means from the laboratory, if it has to move to the land, that's the translation. That has to happen through the entrepreneurship. Next one, please. Waste to wealth. I think microorganisms, plant pathogens can really play an important role, interdisciplinary role. Next one, please. So this is one arecanut husk in Shimoga area. I told you 1 hectare area will produce about 12 tons of husk. If you leave that husk on the road, it's a nuisance, because once the rain is there, it will block the drainage. If you burn it, it will be environmental pollutant. So one of the best things the Shimoga scientists and the pathologists and the microbiologists together, what we have done is, we identified the microbes growing on that, isolated and purified. Next one please. These were the ones, and the consortium was prepared and sprayed over. Next one. When it was sprayed, we could find, otherwise left to the nature, arecanut husk will take 2 years to degrade, to become compost. But when you use this consortium, we gave it to the deputy, DC of Shimoga, that we will be freely supplying it. You make a compost pit of 5 acres or 10 acres, because that compost is now sold for 10 rupees per kg. That is the value of the compost, because it can degrade in less than 5 months. Next one please. So this is the result of the micronutrients. You see, total nitrogen, total phosphorus, calcium, magnesium, sulfur, iron, manganese, and CN ratio. Today, farmers in arecanut are crying because they are getting disease. They are getting disease because the soil is deficient in all this nutrition, and the plant has lost its immunity. If the soil is deficient, if the soil is facing anemia, crop will face anemia. If the crop is facing anemia, human will face anemia. That is a cycle. So that is where this is important. Next one please. From the same compost, a farmer produced 645 kgs of mushroom from 1 hectare husk. 645 kg. Per kg 400 rupees. You can imagine the income. Then when he dried it up, he sold it for 4,000 rupees. That means he added value from 400 to 4,000 rupees. Next one please. So I don't want to touch upon, Madam has already dealt in detail the omic research, what are this application, precision plant pathology, biosafety, diagnostic, genetic engineering, and next generation sequences, and nanotechnology. Next one, please. All these are the interface. One example I wanted to cite, artificial intelligence and machine learning. One of the best example is maize rust caused by Puccinia sorghum. The data accuracy, the size, the pustule count, the volume of spores, everything was converted in a testing using a unit convolution neural network, and then they were able to give a quantification and prediction through this neural network. Next one, please. I don't want to touch upon, next one, please. Lot of tools have been already discussed morning by Dr. Celia, Dr. Reddy, and also Madam. Next one. Era of fungal genomics. Next one, please. We have already, I think, proteomics, the numbers have been increasing every day, it increases. Next one. I just want to quote one example of resistant variety of wilt, pigeon pea wilt, against Fusarium udum. We took almost between the resistant and the susceptible, 141 differentially expressed protein spots, and 12 were successfully characterized using Malditoff, and 2 defense-related proteins were identified. Next one. This is how you can see the R56, and this is in the susceptible variety S41, down-regulated. Next one. And we could find R56 was nothing but the pathogenesis-related protein which was involved in defense. Again, S41 was visible in susceptible, which was not offering the defense. Next one. In the biocontrol, I think, as I told you, we have not achieved the success because ground reality in biocontrol is demonstration at the field level. For several reasons, we are not able to show it, but definitely nowadays regulatory reforms are being done because the quality products are not available. That's why regulatory reforms are being done. Consumers demand for healthy food and safety food, and there is an increased investment on R&D of biopesticides, and the farmers' education and support is also given nowadays going towards global market access. If you look at the European market today, they have declared a program called as green. Under that green, I think 50% of the pesticides, that means that are going to be released by 2030 will be biopesticide. Even US Environmental Protection Agency, in the last 1 or 2 years, 20% of the pesticides released are biopesticide. The Asian countries like, of course, India, 1968, we have a CIBR rule, but still the bioproduction is given some importance. But countries like China, in 2015, they declared it as a green pesticide. That means they don't want to have any, they want to have a zero pesticide, chemical pesticide. So these are all the indicative that the technology is coming up in microbiology, microbial technology, and the world is going towards demanding consumer is demanding healthy food and very food free from pesticide. So also the regulatory reforms are being taking place. So we find a bright light for biocontrol also. Next one. Yeah, I think we have traveled 3.5 lakh kilometers searching for moon, searching for Mars. Hats off to them, but we don't know what is there in the 6 inches below the soil. This is the beauty of 1 gram of soil. You can see it is a question of $1 billion. I think the startups, we started startups back in 2016 with 450. Today it has crossed 10,000. That is an indication where the bio inputs have become most important. Next one, please. So trichoderma, probably wider adaptable. It has application in waste utilization, pharmaceutical industry, compared. Also, it's one of the most important and resilient one for climate. It not only biocontrol, it has got the growth promotion also. Next one. So that was during the pre-genomic era. With the genomic era, when the sequences are done, whole genome sequence, today we are in a position to say the secrets, unravel the secrets of biological control. As many as 200 genes are being identified as mycoparasites. And there is a possibility of strainal improvement through crossing. And it has become a very vast reservoir of cryptic secondary metabolites. And there is also possibility, these are the candidate genes, transgenes from trichoderma against pathogens, against thermotolerance, against salt, against osmotic stress, cadmium. So possibility of trichoderma evolving into a bioagent in the next 10 years could be much more than what it was. Next one. So these are all, there are 200 bioactive molecules, volatile antibiotics available. Next one. So similarly in Pseudomonas, antibiotics, hydrogen cyanide compounds, and siderophores, these are all are possible not only through even cell-free suspension can be also used. Next one. Just the formulations. Next one, please. So we went in for field evaluation using IPM. Next one. IPM, of course, in India, cotton is the success story. Of course, basmati rice in northern India, pigeon pea, chickpea, and vegetables. But again, the demonstration and field ground data, low quality inputs, and IPM as such is a very knowledge intensive. It's not easy, as we say here. It is even a pheromone trap is a knowledge intensive. So that's why, next one please, we conducted, this is one of the Green Revolution belt in Upper Krishna project area, where we find chili hybrids and heavy fertilization. And as a result, pesticides are sprayed day in and day out. Almost a cocktail of fungicides. What you end up at the end of the day in Ballari and Raichur, 40 sprays per season of chili. And next one please. So the production cost is high, protection cost is high. Dr. Reddy's viruses are plenty, as many as more than 2 dozen viruses. Next one. So that's why ICR gave us a demonstration trial to conduct for 10 years.
Please.
I'm concluding now.
Yeah,
Next
please
one.
conclude.
Next one. So this is the demonstration we conducted. Next one, please. So we started with 5 acres. We went up to 500 acres. Next one. So this is the result of IPM demonstration. As you can see, this 7 years data is there. But in 2009-10, there were number of sprays were 8. The number of pesticides, biopesticide spray were 5, as against 22 in the farmer's field. And total harvest was 3.55 tons, as against 2.6 ton in the farmer's field. And total gross return was 2.4 lakh with a benefit to cost ratio of 1.582. So that's the indication of when you can bring down, because that was the intention of the ICR, that you should bring down the number of pesticide spray and increase the number of biopesticide spray. Next one. So I just wanted to say that 4% or 5% in coverage, 96% has to come from, because we don't have a quality inputs. If the quality input has to come, it has to be mass scaled by entrepreneur, agri graduates, or science graduates who are trained in bioentrepreneurship. Next one, please. So I just gave the example of entrepreneurship by Krishnamurthy Yella. He actually agri graduate, master's degree in plant pathology, entered into vaccination, Zika virus vaccination, and even hepatitis virus, then including the COVID virus. And the hepatitis B he sold for 10 rupees, which was $70 at US. So that was the entrepreneurship where the entire family is into vaccination. Similar kind of public-private partnership in bioentrepreneurship, biocontrol agent, biopesticide is expected from agri graduate. Then it is possible to cover the remaining 96%, or another 50% will make big difference for food products. So we had the honor of honoring him with the honorary doctorate. The first new university honorary doctorate was given to Krishnamurthy Yella, an agri graduate, for the entrepreneurship. Probably Madam will holler some agri graduate if somebody of agri graduate could show some of this entrepreneurship skill. Next one, please. Today's startup is around, this is how it has spread now. I think Maharashtra, Karnataka, and Tamil Nadu in the forefront. Telangana is also in the forefront. Next one. The last slide, way forward, I think India is, although very ancient in civilization, youngest on the planet. Average age is 29, and almost 65% of the population is around 30 years age. So that's why we have possibilities for agriculture should be an engine, and if at all the government of India says Vikasit Bharat, it has to happen from the farmer's field. Taking agriculture as a power of engine to move, then only it's possible. I think our GDP is now $3.5 trillion, and probably maybe we are the fourth one. We might beat Japan in another couple of months, but those are all the possibilities, provided the biocontrol, biopesticide, and the agri-industry flourishes. It's possible to attain. Next one, please. I just end up, this is what Bharat Ratna Swaminathan says, that the countries which produce the grains, the future belongs to the countries which have grains, not the guns. If you don't have to buy the guns, you can purchase grains but not the guns, right? So productivity has to be enhanced by 40%, hunger by 30%, carbon footprint by 20%, and there is a role for plant pathologists. Key challenges are there. We can be a partner in that. Next one, please. I acknowledge the ICR, DPT, and my students and scholars and organizers, especially Honorable Vice Chancellor Dr. Sharda Devi for inviting me, and Dr. Josna also took a lot of trouble in making me to come over here. And I had your darshana and also the darshana of Venkatesha. Thank you very much for your patient listening. Thank you very much.
Scintillating lecture, sir covered past, present, and the future of plant pathology. So next, sir, any questions? Sir, any questions?
Take another 1 hour if question answers are allowed. Because he has ignited everybody. People will start asking questions, it will take another couple of hours.
So next, the plenary talk, Prospects for Sustainable Plant Disease Management in Vietnam, by Dr. Nguyen Dac Khoa, Vice President and Secretary General, Vietnamese Phytopathology Society, Can Tho University, Vietnam. I request S. Reshma to read a brief biodata of Dr. Nguyen Dac Khoa.
Now I would like to introduce Dr. Nguyen Dac Khoa. Sir obtained his BSc in Agriculture at Can Tho University, Vietnam in 2000, MSc in Molecular Biology and Biotechnology at the University of the Philippines, Los Banos, and the International Rice Research Institute, Philippines in 2005, and PhD in Plant Pathology at the University of Copenhagen, Denmark in 2011. Sir has been appointed as an Associate Professor and Principal Lecturer at the Institute of Food and Biotechnology of Can Tho University since 2020. Sir has led his plant pathology group since 2011 with more than 170 members, including 7 researchers, 6 PhD, 34 MSc, and 131 BSc students, producing 2 PhD, 31 MSc, and 125 BSc graduates. Sir was concurrently assigned by the Can Tho University to be the head of Research Management Unit of the ODA loan project funded by Japan, entitled Can Tho University Improvement Project from June 2016 to July 2025. Sir has several significant outputs in research, such as 2,143 peer-reviewed papers, 97 books, 69 new technologies, 297 graduate degree holders, 98 conferences and workshops, 56 short trainings, and 2,810 trainees. Sir is currently the Vice President and Secretary General of the Vietnamese Phytopathological Society. The major research areas of Sir include identification of pathogenic and beneficial microbes, control of plant disease using antagonism, control of plant diseases using induced resistance, and investigation of pathogen population diversity for durable deployment of genetic resistance. Sir was conferred a recognition award for excellent research achievements in soft rank province by the chairman of People's Committees of Soft Rank Province in 2019. Sir was then recognized as a distinguished scientist of Can Tho City by the chairman of People's Committees of Can Tho City, Vietnam in 2023. Another recognition award was conferred to Sir by the President of Central Council of Vietnam Union of Science and Technology Association, for his excellent job performance to develop the Vietnam Union of Science and Technology Association during 2022 to 2023. In addition, the President of National Education Union of Vietnam conferred Sir a recognition award for his excellent achievements in research and technology transfer. Internationally, Sir was awarded the Sarkar Regional Professional Chair for academic year 2023 to 2024 in the field of plant pathology by Director of Southeast Asian Regional Center for Graduate Study and Research in Agriculture, for being an outstanding Southeast Asian expert in academics, whose achievements have made great impact, accelerating transformation through agricultural innovation. Now I would like to invite Sir to present his plenary talk.
Thank you for your very nice introduction. We are much behind the schedule, right? Delta Vietnam, which is the major agricultural productions of the country, to you, and I would like to hear from your comments and input during my presentation so that we can learn from your Indian Phytopathological Society to bring home. Does it work? This is the year screen. This is my society. It was established 2001 with 21 branches, more than 1,000 members. And as I told this morning, that we successfully organize the conference annually every year. We have the national conference of our society, and we publish the proceeding annually. And so far we have published more than 600 plant pathology paper of our society with our peer review. Two important events. There, it's good, thank you. Two important events that I would like to take this opportunity to announce that our coming national conference will be organized in my university, Cần Thơ University in Cần Thơ City, in the 16 to 19 July 2026. And this is our honor to invite you to attend. Just write me an email, then I can follow up with that to invite your society to come with us. Another bigger conference is the Asian Conference on Plant Pathology. We will host in 2027, also in July 28 to 31st of July 2027 in Gia Lai province, which is a very nice place for tourists as well. So we will publish this one on our website for you to register to attend. My university, Cần Thơ University, is one of the strongest university in agriculture of Vietnam. And those are informations about my university, so I'm going to skip this part to go straight to the Mekong Delta. So if you fly to Vietnam, there are two places you can fly to. It's Hanoi and Ho Chi Minh City. And from Ho Chi Minh City, if you go down to the south 170 kilometers, you will come to Cần Thơ City, which is the capital of the Mekong Delta of Vietnam. And as I told you, this region is the major agricultural productions of the country. This area, and we have several crops, aside from rice, which is the major one. We have fruit crops, and if you come during the conferences I just announced, which is the fruit season in summer, you can taste a lot of fruits there with affordable prices and very fresh. Talking about plant pathology, I'm going to start my talk by the disease triangle. So a particular disease only occurs with the presence of all three components: a susceptible host, a virulent pathogen, and favorable environmental conditions. So in this using this principle, if we could remove one of the three components, theoretically we can control a particular disease. So for the host, we can manipulate the host by genetic resistance using traditional breeding or genetic modifications. This morning we hear about genetically modified organism or crops, and we hear about gene editing, or recently people still stick to traditional breeding using MAS or marker-assisted selection, which is more acceptable to everyone. The new areas that my groups work on is induced resistance, where we use different inducer to activate plant immune system. For the pathogen, if we would like to remove the pathogens, we can use chemicals or biological control agent. And in this talk, I will focus more on a biological agent. And of course, we need to offer good environmental condition for the crop, but not for the pathogens to develop. And all of these three components need to be integrated into the IBM system. We use this principle to control plant diseases in Vietnam. And what I'm going to talk in the following slides is our practical work and our current research in my group as an example. These are the work that my group has done. For genetic resistance, we work on rice bacterial leaf blights. For induced resistance, we work on rice blast, rice cyst blights, rice bacterial leaf blights, and currently I have a PhD student working with Longan fruit rot. Using antagonism or biological control agent, my group works on rice, on shallots, on sweet potato, on rose and cucumber. And rice is still my major crop to work on so far. I work in the Institute of Biotechnology, so in the Department of Molecular Biology. So I use these two tools to assist our group to work on plant pathology. First, we can use my molecular biology and biotechnology to understand interaction among the host and the pathogen, and then in environmental conditions. We can use molecular biology for detection, identification, and characterization of the pathogens that we have heard from previous lecture. We can use for disease diagnosis, for breeding and deployment of genetic resistance, for induction of plant immunity, screening for effective disease control agent, understanding pathogen evolution, adaptation, and resistance, and many more. So I'm going to talk about pathogen detection first. We use molecular tools to detect some monasterized rice bacterial leaf blight. This is an interesting pathogen because the population is vary a lot. I'm going to show in the following slide. Aside from that, we have heard a talk about plant quarantine, and we also, in my group, we work on rice cyst-borne fungal pathogens and bacterial pathogens. Some of them we can use morphological characteristics, which is much simpler, but many of them we could use molecular tools for detection. For cucumber, we work on Coletotrichum. This is a nice story that finally I got an award from the local government. Shallot. At that time, this is a very caused by only insect, but also pathogens. And the way people apply that is they just apply insecticides, but not for biological control agent and plant extract. And we was able to control the pathogens at that time to help the farmers. So we identify these pathogens and we work on different pathogens. Then we offer the farmer biological control agent using living microbes and plant extract so that the farmer there don't have to use chemical anymore. So that helped a lot. For sweet potato, this is a major crop in another province which give the farmer a lot of money by export. We export sweet potato to China and to adjacent country, and this is a high value crop. And it's got Fusarium. Slide is about only identification of pathogen or characterization of pathogen. I'm gonna skip, we also work on roads. Currently, we work on long-run food growth, and we identify Lasiodiplodema theobrome, which is a newly emerging pathogen in Vietnam in long run. My PhD student will defend 2 weeks more about this one. That is identification or detection of pathogens. Let's talk about control of plant diseases. I'm going to talk about genetic resistance, induced resistance, and antagonism. For genetic resistance, I work with this pathosystem since I was in Erie. And this is an interesting pathogen because the pathogen populations vary through time. The pathogen is able to adapt to the resistant variety that have been deployed by the farmer in the field. And after some time, resistance will be broken. It's not durable. People have identified more than 40 resistant genes, and in Erie, people make gene pyramids, which is they transfer different genes in one variety to deal with different races of the pathogen, but still it's not that durable because of this co-evolution pathway of the host and the pathogens. So you have read the paper by John and Dango in 2006 about co-evolution, and read is always a race. We said that the pathogen and the breeder was actually the pathogen and the host. They have co-evolution as we have to deal with, and the microbes always faster because they have similar genome. We found some newly emerging races in Vietnam. I did with the word collection in Erie, and then we see the variation, and I used that tool to study the local population, and we saw some newly emerging races as well in Ixola. Using genetic resistance is advantages to farmer, similar, but there are some disadvantages that the farmer told us. The first one is we need to understand pathogen population structure before we could deploy a particular resistant gene or gene pyramid. Resistant durability is not that high, so after some years of deployment, it's not there anymore. Seed supply. So normally farmers are dependent on the company who supply the seed. And using genetic resistance that may impact on the indigenous germplasm and the ecosystem as well in case of GMO. So those are concerns. After genetic resistance, we talk about induced resistance because all organisms have their own immune system, immunity. So for plants, they have 3 classes of response, defense responses. The first one is the physical barrier. They could establish papillae, which is the special structure against fungal pathogens. They could establish lignification, scalar accumulation. These are special structure against the penetrations of fungal pathogens. Chemical barriers. Plants could produce toxic substances against the pathogen. They call phytolactin, phytoanticipin, or antimicrobial compounds. And finally, plants also do hypersensitive response of programmed cell death to colonize the infections of a pathogen, not to spread around the other adjacent tissues. So if we understand this one, we try to stimulate this kind of thing so that the host plant can protect itself. These are some pictures showing the defense responses that I just mentioned: papillae formation, lignification, callose accumulation, and productions of secondary metabolites. For example, in this case, against fungal pathogen, plants could produce beta-1,3 glucanase, which could degrade the cell wall of fungi. And this is the hypersensitive response. So, still, all plants have these kind of defense responses. These responses come on time and strong enough or efficiently. So far, people have identified different chemicals, microbes, the VGPR also can do, and plant derivative, which could activate or elicit the immune system or defense responses to come on time and efficient against the pathogen. I started my work with induced resistance with my supervisor. He passed away already, the one on top. And we screened for different chemicals at a time. We used chemical elicitor. And we found one chemical which is not toxic, which could stimulate the immune response against rice blast. We give these chemicals for free to farmers, and they were very happy to apply, and they follow. But finally, when we transfer this technology to the company to produce the product, so farmer don't buy anymore. So there's different reason behind that one. Why we give for free, farmer use, and when we sell it, farmer don't. So if we have more time, we can discuss on that. The most important point is that for this kind of strategy, farmer need to do in advance, and I'm not seeing any obvious damage they don't do. And induced resistance is the one that require farmer to do in advance, to apply in advance. That is the most difficult points of induced resistance. Using that principle, I did successfully found a plant extract which could stimulate rice response against cyst blight caused by Rhizoctonia solani. So those are dependent-related genes in rice against Rhizoctonia solani, and when we apply the plant extract, we see the upregulations of these genes in the rice tissue. And we also pick up one, for example, beta-1,3 glucanase. We see upregulation of these genes. We also see elevated level of this enzyme in rice tissues some hours after. I supervised one master student in Denmark to do fractionations of this plant extract, because when I did that, the plant extract was a cocktail, was a mixture of everything. And we tried to identify the active compounds inside the plant extract, and then we have not finished this one, and I had to return home. But one of my PhD students finished this one for another pathosystem that I'm showing later. This one, plant extract against rice bacterial leaf blight. So we also use the same principle to work on this pathosystem, and then we could prove that this plant extract, this one plant extract, Kalanchoe pinnata, the live plant, could stimulate defense responses of rice plant against bacterial leaf blight. And my PhD student identified active compounds already. So what we will do is to study the structure of this and try to synthesize this structure, because making plant extract, we don't have sufficient amount for large-scale application. So our approach is to identify the compounds in plants, study the structure, and synthesize it chemically. The disadvantages of using induced resistance is that people need to apply in advance. So that's the most difficulty. No immediate effect. And for induced resistance, you don't expect to have no symptom. Symptoms are there, but fewer number and smaller lesion. That is induced resistance. So still, you see the disease, but it doesn't damage the crop, the yield. Sometimes you need booster application. You need to remind the plant because the plant don't remember. And it's just like vaccination, and farmers are not happy about this because they have to repeat application several times. Variable performance. This is the weakness of biological control. When you apply in the field, the effects will vary. And when farmers see different effects, not as that as they expected, and they stop using it. Only for one time, they don't trust anymore. So that's also a barrier. Energy consuming process, of course, when you use something to activate the defense response gene, it consumes ATP, it consumes energy, and therefore it will decrease the yield of the crop. But we trust that although it decreases the yield of the crop, it's still better to let the pathogen damage the crop, so it's still okay to apply in this system. Metabolic burden sometimes stimulates the plant to produce a lot of secondary metabolite which are toxic to the pathogens, but this is also a burden for metabolism of the plant as well. Signaling crosstalk, this is in more detail about induced resistance, because induced resistance also works against insects. And when you stimulate the immune system, again the pathogen, you deactivate the immune system, again the insect. So it's crosstalk and it's block each other. So this impact of indigenous rhizome microbiome is just new that people find that when we use something to activate the plant, so it affects the symbiosis microbe in the root system. And of course, biological control requires user knowledge and skill. This is also a big area. So let's turn to biological control agent using antagonism. We base on the interaction among organisms, and of course we use the antagonistic interaction to screen for different biological control agents. We work on rice yeast blight, we work on rice bacterial blight, and currently we work on rice bacterial barnacle blight. Bacane as well. Charlotte Becerot, I told you in the blind village. We also use plant extract. Our group work on two different fields, antagonistic soy bacteria and plant extract, indigenous plant extract. Sweet potato tuberose, rose leaf rot, cucumber anthracnose. So these are pathosystems that we use biological control, and it has a disadvantages as well. No immediate effects, variable performance, as I told you, impacts of chemical treatment. When you use biological control agent, you cannot apply them together with chemicals because the chemicals may kill them. And normally the farmer do so. We ask them not to do so, but in practice they do so. That's the barrier. Impact on indigenous microbial population. When you spray something that are useful into the ecosystem, it will compete with other microbes as well. So currently we try to convince farmers to go for biological control to spray useful microbes into the ecosystem, but be aware that when you spray until a certain level, it will compete with all other microbes in the naturally balanced ecosystem. You break the balance. Pathogen health action, people don't trust this, but it's there. When you apply something, it's not only chemical causing resistance of the pathogens, but when you use biological control agent, the pathogen also fight back to be resistant. It's occurred. Involvement of new pathogens by horizontal gene transfer, something you feel useful, but we never know after some time it's become harmful. That's microbes, because that's too simple in the genome. They can shift anytime, especially via horizontal gene transfer. High cost, studying it takes time. Storage takes time. Actually, most of the time, the difficulty is how to store the biological control agent in a bag for commercialization. If you have bacillus, that's the gram-positive with endospore, that's fine. But if it's a gram-negative bacteria, then it's much more difficult to store for at least 12 months or 18 months, at least, from the manufacturer to the farmer. This requires user knowledge and skill as well. So these disadvantages explain us why biological control are eco-friendly, but until now, it's difficult to get in line. To summarize, to control a plant disease, you can work on the host. By genetic resistance, you can induce its resistance. You can kill the pathogens by biological control, and these things need to be integrated in the IPM together with the cultivation activity. One point this morning, Madame said that the stakeholder linkages, farmer, industry, and scientists. I would add more in government. This is the actual condition in Vietnam that we need the four of them working together before we could figure out an effective control in general in agricultural production. We cannot miss government in here. So in our practice, we need these four partners or stakeholders. I would like to thank my supervisor and my group I think I have to thank Dr. Bartol because without him, I'm not here. We met in Vietnam in a conference, and after this shake hands, I come here and we become good friends. That's yesterday. Thank you very much, Dr. Bartol. And we are doing sustainable agriculture for now and into the future. It's not easy. Thank you very much for your time.
I have one or two questions still we have. We have less.
I am forced to ask you a question. In fact, I find Vietnam is not a member of ITPGRFA, Plant Treaty. You have some knowledge about ITPGRFA, International Treaty on Plant Genetic Resources for Food and Agriculture. You are a member of, you are a party to CBD. Vietnam is a party to CBD, Convention on Biological Diversity.
Yeah, that's 1992, right?
Yeah, that's right, 1992. You know, that summit also happened, and it is a development of the global South's diversity-rich countries. You have tactically handled that situation. I have all the congratulations for the Vietnam. I tell you why. I think if I can take a minute more. You are not a member of party to ITPGRFA, where ITPGRFA treated all the diversity-rich countries very shabbily. You know, the global north exploited all the. It is a ITPGRFA is a multilateral treaty, where actually the parties, all the parties, about 190 countries or so, are the party to the ITPGRFA, where the genetic resources of all these diversity-rich countries are controlled by, the ITPGRFA, and whichever country wants genetic diversity, they can access very freely. Being a party to CBD, that's again an international treaty. You have rejected ITPGRFA because that is a multilateral. You don't want to give access to the global north that actually took the material from the international gene banks. I mean, for example, there is international gene banks are there among all the CJR institutes, from where the member countries of, it's all FAO control, member countries can access the germplasm without having any restriction by the national governments. So what exactly happened is, they have actually almost 7 million accessions of germplasm has been transferred from MLS. No single dollar has been paid back to the communities of these diversity-rich countries. That was the damage done to the diversity-rich countries. Whereas CBD is a master stroke. Signing CBD, it is actually, it protects the member countries. If somebody accesses material from your country, the user, whether it is a country or an institute, a seed company, they will have to pay back the benefits. A part of the benefits have to be shared with the original provider. Either it could be a community or a country. So you have a master stroke you played. I'm really very appreciative. I was just wondering how Vietnam very tactfully did not sign the ITPGRFA. This is the information I wanted to give you. Thank you so very much.
Thank you. I don't really know that, but we joined the Nagoya Protocol.
Yeah,
and currently...
Yes, Nagoya Protocol is a part of CBD.
Yes.
That's all rules, for sharing the germplasm. And, ITPGRFA had you signed the ITPGRFA, you would have lost like India lost to the, the users of the global north.
Thank you.
Thank you for sharing your area of work. It is good information for all of us. Thank you.
Thank you.
So the preliminary session was completed. So we will start technical session 1. The theme of the technical session 1 is harnessing phyto-microbiome for sustainable crop health. So for this technical session 1, I request Dr. M. Krishna Reddy sir, former Principal Surgeon Head, ICAR IHR Bangalore, to act as a chairman for this session. I request Dr. Reddy sir to to come on to dais and please have sir. And I request co-chairman Dr. V. Prasanna Kumari Madam, Professor Head, College of Agriculture, Bapatla.